I think we are ready to start. If anyone's outside and you can hear, if you'd like to come in. Okay. Well, welcome everyone. I can't see with the bright lights in my face, but it looks like we've got a nice turnout this morning, and welcome to SAIAB's Research Day, where we get to sit back and enjoy listening to the research that you guys, the students, have conducted over the last year, over the last 2 years. Perhaps you're coming to the end of your PhDs, end of your masters, maybe you're in the middle and have some exciting results. And I know that we're all very excited to hear what you have to say. I think these kind of research days are such a celebration of who the Institute is, of what the Institute is, and where you get to showcase your efforts, but also your supervisors' efforts, the support team's efforts. You can't be where you are as an individual. You're here because of the support you get from people in the front office, people in finance, people in HR. Welcome, guys. And I think to always acknowledge that who is around you, who helps you to be who you are is, oh, I'm moving this podium. Sorry, I'll fly off the stage. Yeah, it's always it's a good thing to do, is to acknowledge those people around you who supported you and pushed you through the way. Perhaps even a partner, a parent, a friend. And as you come up here this morning, just take a minute to think of who you are and how you got here. And we wouldn't be here today if it wasn't for the amazing efforts of the organizing team, who have done, I think, an incredible job of getting this together, of getting a really packed program together. We've got a hybrid event this morning, and that's no easy task. So thank you so much to Albert's team, and I'm going to read everyone's name out. To Tolwana, to Yonela, Tadiwa, Martinez, Devan, Melissa, Shiluva, Nkululeko, and Esihle. Thank you so much for putting this morning together. And I'm sure we are not going to have any hiccups. It's going to go smoothly. No hiccups. And please just remember to stick to time, to listen to the chair of the session. And yeah, and I think with that, I will hand over to our first chair, who's Dr. Nompumelelo Baso, and she will be here to introduce our keynote speaker. So thank you very much.
Morning, everyone. As Julie has already said, my name is Nompumelelo Baso. Many of you might not know me, but I'm based in Joburg. I exist. So the first person that will give us the talk is Chieza Kenneth. Sorry? Oh, sorry. Keynote. Oh, sorry. Keynote address first. I thought I wasn't sharing the keynote address. Oh, I feel special. Sorry, Dr. Manda is coming to the stage first to introduce the keynote speaker then.
I see that we're off to a flying start. Good morning, everyone. We are More and Mancheruni, Abusheni, Dumela. So, as she has already mentioned, my name is Mandla Magor, and I am an instrument scientist at SAIAB. So, my job for today is actually quite easy, because the man I'm about to introduce is very familiar to SAIAB, and it's a very prominent name within the aquatic research space. Professor Alan Whitfield has a PhD in estuarine ecology from the University of Natal and a Doctor of Science in estuarine ecology from Rhodes University. Alan first joined the JLB Smith Institute for Ichthyology in 1978, and he rose through the ranks and became the chief scientist at SAIAB, a role that he served with distinction until his retirement in 2018. Now, Alan's research interests mainly fall within estuarine ecology, with a keen interest in the fish which inhabit estuarine systems. His work has covered topics such as the feeding ecology of these estuarine-associated fish, classification of estuaries, fish larval ecology, population dynamics, and the importance of river flow in the functioning of estuarine systems. Now, over the years, Alan has been very kind to also train several postgraduate students, including at MSc and PhD level. And this list also includes yours truly, myself. And Alan is also a recipient of the prestigious Sussex Gold Medal Award that he got in 2015 for his contribution towards research. Alan has published more than 200 scientific works. And even in his retirement, Alan remains productive. He continues publishing, and his work also includes recently publishing a book on the Neisner estuary. Now, Alan, for today, is going to be covering a theme around safeguarding Africa's estuarine resources. And I am very much hopeful that the younger audience here will benefit from hearing a talk from someone like Alan with all of his experience and his accomplishments. And this will inspire you to push on forward and develop yourself and become a leader in your own field. Ladies and gentlemen, it gives me great honor and pleasure to welcome on stage my MSc and PhD supervisor, Professor Alan Whitfield.
Thanks, Mandla, and welcome to everybody here this morning. Yeah, it's interesting that I was looking through the program for today, and when I joined the Institute in '87, there were mainly taxonomists, systematic people. I was one of the sort of first ecologists to be employed. And it was really good to see the group that Mandla belongs to. It's called CLAPI. It's a group on fish systematics and taxonomy. And to me, it's wonderful to see that that discipline didn't disappear, because there was great concern in the latter years, last couple of decades, that we were just losing our taxonomists and systematists. They weren't being replaced, and it wasn't being picked up properly. And it seems like the younger generation are in fact very active in this field, which is great to see. The other thing I would like to just point out is that I joined the JLB Smith Institute of Ichthyology. It was only fish, and then we changed our name about 1999, 2000 to the South African Institute for Aquatic Biodiversity. And one of the concerns was, how do we transform from a fish institute, as it were, into a broader aquatic biodiversity institute? And looking through the presentations that are going to be made today, it strikes me that this is the way it's happening. In other words, there are such a diversity of student studies that are not on fish, but in a broader aquatic field. So thank you to all those students for broadening SAIAB into a truly SAIAB organization, which wasn't the case when, as I say, our name was changed, but we were all studying fish only. So with that, let me just check if I understand how this works. Okay, there we go. I'm going to be talking about safeguarding Africa's estuarine resources, but I'm going to, in 20, 25 minutes, you couldn't cover that topic at all. So I'm going to focus in on estuarine fish resources, because that is something I know a little bit about. And even then, I'm going to only be covering it fairly superficially, but I hope I can convey some messages to you that are meaningful. That estuary you see in that photograph is the Influentiana estuary, which is just north, it's in the Eastern Cape, it's just north of the Mbashi, south of the Umgazana in Eastern Cape. Now that estuary doesn't need safeguarding. It's as you can see, in a near pristine condition. Estuaries really, they can look after themselves if we're not putting pressures on them which are abnormal. And when I say abnormal, I mean hugely abnormal. And I'm going to show you some examples from Africa, which is abnormal. But if you looked at that picture now is taken in an interglacial, in other words, it's a warm climatic period. But if we went back 125,000 years, which is the last interglacial in the world, including Africa, that picture would be almost exactly the same, because the sea level at that time was almost the same as what it is now. In between, of course, the sea level changed very much. So if that photo had been taken 20,000 years ago, there would be no estuary at all. There'd be no sea in the picture. There would just be a river. So the Intlotjana River would have been there, but no estuary. And the estuary and the communities living in the estuary, they adjust with these massive changes in sea level and the changes that occur in these systems as a result of global and climate change, or climate change in particular. Now, the use of fishes in estuaries in Africa is very, very limited. And the earliest known use of estuarine fishes is from the Elands Bay Cave, which is on the west coast of South Africa, next to the Verloren Vlei estuary. So that cave actually looks out. It's right on the coast, but it looks out over the estuary of the Verloren Vlei. And the digs that were done in that cave, looking at the stratigraphy by the University of Cape Town scientists, they discovered that about 10,000 years ago, which is when the sea level rose. So 20,000 years ago, the sea level was way down. And then 10,000 years ago, it was roughly where it is today. And the people living in that cave, probably ancestral San people, were feeding extensively on white steenbras. Okay, white steenbras, which is a fish that uses estuaries as nursery areas. And the bones of the fish that they found were indicative of the size of white steenbras that you normally find in estuaries. So you get the juveniles and sometimes sub-adults in estuaries, but the adults of the white steenbras are in the marine environment. No adults in the cave, but lots of juveniles and some sub-adults as well, because they were able to determine the size. They also used Zostra eelgrass from the estuary, probably as bedding. They found that in the cave as well. So estuarine resources were being extensively utilized as long ago as 10,000 years ago. If we want to come to the more modern use of estuarine resources, these are fish kraals, Tsonga kraals in Maputoland. The Amatonga people have been using these fish kraals for hundreds of years. I put there 500 years. It could have been way beyond that. But it was a traditional use of fish kraals by the Amatonga people, and it was a sustainable fishery until about 1990, when the number of fish kraals more than doubled and illegal gill netting started taking place, and that fishery is now in serious, serious trouble. The really big impact on estuarine resources came with the European settlers in South Africa, both recreational, particularly recreational fishermen, or fishermen just fishing for fun. And the harvesting of these fish, even in the 1800s, was intensive. So you're looking at the late 1800s in the Swartkops estuary, which is just nearby here. There was a big complaint from the fishermen that the fish catches were declining. So they got the one and only marine scientist in South Africa. In the center of that picture, he's holding up an adult white steenbras. There are no more adult white steenbras there. So he was coming to a system that had already been significantly pressurized from a fishing perspective. And that was one seine net catch. Now, what it indicates is that the pressures on the fish resources of our estuaries, and this doesn't only apply to South Africa, it applies elsewhere, and I'll give you examples just now, goes back more than 100 years, but it really became intensive, as I say, in the 1900s, which is when that picture was taken in 1915. So the impact on fish in terms of fishing has been significant over particularly the last 100 years. And in more recent times, there's been a growing use by subsistence fishermen as well. So the two pictures on the right with a recreational fisherman and a subsistence fisherman, and the fish are getting smaller. So the lady on the left, it's a picture taken a lot longer ago, and obviously the fish sizes are getting smaller and the fish abundance is getting smaller. And people don't realize that in South Africa alone, there's over a million, between 1 and 2 million recreational fishermen. Each person catches, let's say in a year, 1 fish. That's 1.5. If they catch 10 fish in a year, you're starting to talk in the tens of millions, which is a huge pressure on fish resources in estuary. What about Africa? Okay, I went to Angola, this is about a decade ago, to the Kwanza estuary. On the left-hand side there, you'll see a picture of myself with a fish, a tarpon, a giant tarpon in the background. And the tarpon don't grow that big, but they obviously advertising. These are international fishermen who come from all over the world to fish for these giant fish. And tarpon were one of the fish they would go for. The other fish that they do catch is the mangrove snapper. In the lower part of that picture, you'll see mangroves in the Kwanza estuary. And that photograph of a mangrove snapper was one of the visitors at the camp at Kwanza had caught that adult mangrove snapper. And those fish are relatively abundant, or were relatively abundant. I don't know what the current status is now, but at the time there was huge concern that illegal gill netting. On the right, you see a little boat with a monofilament gill net. They were being increasingly used, and obviously those big fish were being taken out and sent to Luanda for fish restaurants and that. And there would be a decimation over time. So the big ones would be targeted first, and then eventually it would get smaller and smaller. And we had a conference in Benin, and this was Lake Nakoe, where any fish that survived in that system, you take your hat off to them. The picture in the upper part of the slide there shows you the variety of gear that are being used. And there was a link between the estuarine lake in the bottom left-hand side and the sea. And you can see that they're similar to the traditional fish traps at Cozy. They've got fish traps going right across across that link, which means that the fish can't really get back to the sea in order to spawn, because that's the cycle that you have. The bottom right-hand side, you see a chap there casting with a massive cast net. He didn't catch a single fish in that net, in that very small mesh. So even small fish would have been caught. So what you're dealing with in some of these countries is a total decimation of the fish resources simply by overfishing. There's not degradation of the environment as such, but in terms of fish pressure, it's huge. The one country I think at the moment in Africa where the resources are safeguarded, but by virtue of the low human population and particularly associated with estuaries, is Gabon. So Gabon is one of the countries where, and it's interesting that the recreational anglers go there now. I don't know if they still go to Angola, but they certainly go to Gabon to catch these very big fish. The good thing about some of these anglers is that they actually release the fish. They don't keep them. Although in that case in Kwanza, you saw there that fish was kept. So that's enough about the overfishing. What about the environment? Because the fish are having to withstand pressures of fishing, but they also need the environment to be in a state that will support the nursery function of estuaries. So different fish species use different habitats for their nursery areas. And unless those are conserved and looked after, you will be in significant trouble. And the next couple of slides are from Durban to show you how development can change that completely. So here's a retaining wall along the embankment of the Durban Bay, where the littoral area of the estuary is totally gone. The tide will come in and go out, but there's no intertidal or shallow intertidal area with salt marshes and that in the Bay of Tor. And if you look at the main channel, they've got these huge big channels for the ships to go in, but the shallow intertidal areas, which are such important nursery areas for fish, are absent. So you've either got intertidal sand, high and dry, not good for juvenile fish, they can't go there. They've then forced into the channel where they are vulnerable to predation. What about the estuaries in and around Itigweni, which is where Durban Bay is? You can see Durban Bay is classified there as being totally trashed. Most of the estuaries in Itigweni are in poor condition or highly degraded. Less than half of them are in fair or good condition. The picture, when in 1977, I started a PhD in the Umhlanga estuary. They call it the Umhlanga there, but the Umhlanga is the river. And those of you who remember the looting in July of 2021, the looters, this was that picture in the bottom center is a warehouse, UCL, or I can't remember the abbreviation for it, UBS warehouse, which basically stored hundreds of tons of insecticides, herbicides, incredible poison. And the looters went in there, they took what they could. They obviously didn't take all the chemicals. And then they set fire to the place. And so the fire department came, I don't know if it was the same day or the day after, the 12th of July, which will go down in infamy as a total trashing of an estuary. I mean, if you want to talk about safeguarding. So on the bottom left-hand side there, you'll see some of the fish species and the food web that was present in the Umhlanga estuary. After that fire and all the slurry from the warehouse went down the Umhlanga River and into the estuary, there was nothing. Even the bacteria, I think, probably were dead. And to this day, I'm not sure whether the Umhlanga is ever recovered to anything like what it was before. So we're certainly not safeguarding our estuaries in South Africa. And if you look at that figure on the left, that reinforces the fact that we've got a lot of work to do in order to safeguard and look after our estuaries. Then we move on to climate change, global change. This is the work of mainly Nikki and her team at SAIAB, where you have these additional pressures coming onto the fishes who are already under stress. Because remember, fishes that live in estuaries, they're subjected to salinity fluctuations and that which many species can't survive. And if you put on top of that the climate change scenarios that they're going to be faced with, one asks the question, how resilient are these fish going to be? And can we do anything more, or should we be doing things to safeguard and to conserve and to enable these fish to continue to live in estuaries? And there you see the different impacts coming from the river, from the land, and from the sea. So it's a three-pronged pressures coming onto these species. This is a slide from a publication from one of Nikki's team, Pakama Nodo, and it's one which shows very clearly how changes in salinity, just salinity, can have an impact on your fish community. So the slide on the right-hand side shows you the typical estuarine gradient in the Kariga estuary from the head of the estuary on the right to the mouth on the left. And below those bubbles you see are all the different fish species that are present in the estuary under estuarine conditions. But if you dam the river such that the river flow stops or is very limited to the estuary, you'll get hypersaline conditions, which is shown by the top left-hand cross-section of the estuary. And look at the bubbles below. There's just a total depletion in both abundance and diversity of your fish in the estuaries. So one simple parameter change can have a major change on the fish species in the estuary. The final topic I want to, which is again a human impact on the estuaries that has nothing to do with fishing. This has nothing to do with climate change, but it's got the way we use catchments. And in this case, the Umfolosi catchment is a degraded catchment and sends down huge amounts of silt into the Umfolosi what used to be the Umfolosi swamp. The Umfolosi swamp was drained in the 1950s and canalized by the farmers so that they could farm the Umfolosi floodplain. What they didn't anticipate was that the sediments from the Umfolosi, which are huge, would then go directly into the estuary and cause huge problems in terms of the functioning of the St. Lucia estuarine system. And today, and when I say today, in the last 2 years, the St. Lucia system is flipping from an estuarine lake system into a freshwater lake system. In other words, we are about to lose more than 50% of the estuarine area in South Africa. In other words, we are not looking after the St. Lucia estuary. We are not undertaking the management steps required in order to ensure connectivity. And just to give you one example of how that has changed in the last 2 decades, the slide on the left shows you the 2002 aerial view looking down towards the Honeymoon Bend area of the St. Lucia estuary. The slide on the right is 2019. You can see all the turbid water now from the Umfolosi River. The Umfolosi has been reconnected to St. Lucia because without the Umfolosi River, the engineer said, oh, we'll solve the salt problem. We'll just make a new mouth for the Umfolosi. But then St. Lucia virtually dried out in major droughts. You can't have that. So you need the water, but with the water, you get all this incredibly high silt. And the consequences of that high silt are now that the channel, which is shown in the left hand, is the little bar there, and there's the little bar on the right-hand side. There's not enough exchange possible between the estuary when it's open in order to get salt water up into Lake St. Lucia. And Lake St. Lucia is the key estuarine area. So Lake St. Lucia is now becoming a freshwater lake and is no longer going to perform an estuarine function in South Africa. So what do we need to do to fix the situation? And there are 5 bullets here I've put forward. We need to have appropriate fisheries regulations, and they need to be rigorously enforced. Now, we've got the regulations. There's no shortage of the regulations, and they're good regulations, but we're not enforcing them properly. Ensure that the WISE environmental legislation is fully implemented and rehabilitation is initiated where necessary. I think we've entered the era, and Itigweni was a good example of that, where we need to do rehabilitation. In other words, research should be directed at how do we get these estuaries productive again? Because if they're not productive, they have no use to us and they have no use to the fishes in those systems either. 3, facilitate the creation of a network of protected areas for threatened estuarine fishes to promote the recovery of these species and associated aquatic resources. So we've got marine protected areas, and in many cases, these marine protected areas are working incredibly well. We don't have many estuarine protected areas, and I think that's, again, a topic and a subject that needs more research. Facilitate steps to reduce and reverse the impact of global and climate change on estuaries. Well, SAIAB has got a very active program under Nikki's guidance looking at exactly that topic. And it's a topic that is going to become very big. So we're seeing on the land stage in North America, Europe, all these floods and damaged storms and so on. But at this stage, nobody's really looking at what is climate change doing to estuaries, because the magnitude there could be just as big and as devastating as it is to people on the land. And if it does start impacting estuaries in a major way, it is going to have a major impact on the resources that estuaries provide to us. And then finally, conduct socio-ecological studies in estuaries and communicate these findings to both government and local communities. In my generation, there was a big lack of communication of research, and it was mostly natural science research, not socio-ecological studies. The era of socio-ecological studies is becoming more and more important, where scientists work with communities and communicate information first to the communities and almost activate the communities to take ownership of the resources and the systems that they use. And that is, again, a big area of study that really needs to be done, not only in South Africa, but throughout the continent. And that's my final slide. Thank you very much.
Is it? Oh, cool. Thank you for the engaging talk. I'm very honored to have had this talk from you, luckily catching you before your retirement or like after your retirement, essentially. But yeah, I learned a lot. I'm a botanist by trade, but it was very nice to hear like the background of the fisheries and getting to understand why there's usually those closures of the estuaries and stuff like that. Because I've seen it have firsthand happening. So I'm gonna open the floor for questions now if anyone has some.
Thanks so much, Alan. Always a pleasure to listen to your, I call them stories because they're so engaging. It seems though that the knowledge is there, the science is there. You've got a very well clear alignment of what needs to be done. Rather, maybe in terms of adding more research and more studies, maybe we need to reinforce and strengthen some of the channels between what we know and how do we translate this, which is already very clear to the governments, local governments or the government, the national levels. Thank you.
Yeah, thanks, Francesca. I think there's a role for people with social skills. They need to have scientific understanding, but they need to also be able, scientists aren't always the best people to communicate with government about these sort of issues. Whereas there are people who can lobby and who are effective at enabling government to move in a particular direction and to put in place the sort of things that need to be done. Scientists aren't always the best people to do that. And so there's almost, I think, an opportunity for another layer of so I would say it's in the social science realm, particularly because social scientists are more people orientated than natural scientists as a general rule. In other words, they are coming to it from a social, from a people perspective, rather than from an environmental or from a species perspective of the component. So they need to be able to understand what the scientists have done and how that information, because scientific papers, you cannot give a government minister a scientific paper and say, this says such and such, and this is what you must do. It needs to be done in a very different way. And the skills that are associated with that communication and implementation are actually different to what many scientists possess. So I think there's an opportunity for, I don't want to say quasi-scientists, but scientists who can understand science, who understand government, and can provide the link.
Hi, Alan. Thank you so much for your presentation. Actually, following up from Francesca's point, we are always more orientated towards the scientists and the academics into resolving issues. But how much can these science be translated into works that the community can engage? So a lot of the works that we are probably doing now is engaging more towards citizen science. And maybe there is some form of awareness, but how can the local community itself be more involved into helping engage with the government and the science behind it? So that needs to be translated into something that they can understand and digest and actually be more impactful.
And I think that's where the socio-ecological sciences, which are in the infancy at the moment, probably in a decade or two, they are going to be totally dominant. In other words, there'll be very few natural scientists around, and most of the funding and everything is going to go towards the socio-ecological scientists, who will also engage more closely with communities than the natural scientists did.
Prof, I think we are going to move on to the next speaker, or the first speaker, essentially. And then sticking to the theme of fish and incorporating some technology into it is going to be Chiesa Kenneth. Just a round of applause as he comes to the stage.
Good morning, everyone. I'm Kenneth. I'm a computer science master student at Rhodes University. I'll be presenting my paper on assessing the accuracy of YOLO V8 and YOLO NAS for automated fish detection. The brief overview of the content that I'll be covering today, starting with the brief background. So fish monitoring is important for a lot of things, such as tracking species abundance, studying fish behavior, and it is also crucial for addressing global fish stock exploitation. One of the current methods for fish monitoring is the use of baited remote underwater video systems, known in short as RAVs. This is preferred over the traditional methods as it enables some control in capturing the footage, and it is also considered non-invasive and less disruptive to the fish population. It also generates large amounts of reusable data. Well, the problem with this is that going through all of this data can often be an error-prone and time-consuming process. So a suggested solution for this is automated fish detection. This also comes with its challenges. This is due to the poor visibility of images captured underwater. This is due to a number of different reasons, such as extreme environmental conditions found underwater and the nature and behavior of fish themselves. So there's been recent advancements in computer vision, which has led to the development of object detection models with the potential to overcome these challenges. This brings me to the problem statement. So several studies in the past have made use of these YOLO architectures for the fish detection task. And while earlier versions demonstrated strong performance, there's been little study into the newer versions, which promise further accuracy enhancement. So the aim of this paper was to study the newer versions of this you only look once architectures to address this research gap. So we followed a 3-stage approach to achieving our aim. So we had a data collection phase, and then we trained our models, and then we evaluated and compared them. So for the data set collection phase, we collected 2 public data sets, the Ozfish data set and the Deepfish data set. We regard the Ozfish data set as being more challenging than the Deepfish data set, as it has a lot of small blurry fish, and it also has a high fish density. And then we also use some SAIAB data sets. So we created these data sets in collaboration with SAIAB. So SAIAB provided us with the videos, and then we made the manual annotations. So this resulted in these 2 data sets, and the Tanganyika dataset is the only freshwater dataset that we considered during our experiments. For the training phase, we used the same hardware for all our experiments, and we split our data into a standard 70-20-10 split. So 70% training data, 20% validation data, and 10% testing data. And then we tested all the size variations of the YOLO V8 models and the YOLO NAS models. We did the training for 300 epochs with continuous validation, and then we selected the best model for evaluation on an unseen test set. This brings me to the results. So we did both quantitative and qualitative analysis. So the quantitative analysis uses precision recall and mean average precision metrics to quantify the effectiveness of our model. And then the qualitative analysis, here we ran inference on a bunch of examples. This was to gain insights into the specific strengths and weaknesses of the models. So this is the first experiment, which was on the Deepfish dataset. And we also included a result from a different study by Musket et al., which did the same experiment but using an earlier version of YOLO. So we use this as a baseline to see how the newer versions compare to the older one. Okay, so in this first study and this first experiment, YOLO V8 performed well. It surpassed the baseline, which was set by the earlier version, YOLO V3, while the YOLO NAS model was not able to surpass this baseline. This suggested to us that the advancements made in the YOLO V8 architecture were more effective for this data set compared to the neural architectural search which YOLO NAS uses. This was further emphasized by the 10% difference in precision scores between the YOLO V8 models and the YOLO NAS models. This shows that YOLO NAS made a lot of missed detections during the experiment. This was further validated by the qualitative analysis. So here, the white boxes represent the ground truth, which is the manual annotations, and the yellow boxes represent the model prediction. So here we see YOLO NAS struggled with this image. It detected part of the fish as a separate fish instance. And then we saw that on this data set, YOLO NAS also struggled with complex backgrounds. So here it identified the background as 3 separate fish, but that there was no fish and that it was just the background. And then moving on to the second data set, which I said before is more challenging than the one I just went through. So here, both models were able to surpass the baseline set by YOLO V3, showing the advancements made since then. And in this case, YOLO NAS was even able to perform better in terms of precision. Moving on to the qualitative analysis, we noticed that both models did struggle here, but YOLO NAS was a bit better at detecting the smaller fish. We see this here again, both models struggle. So all the white ones, they did miss those, but YOLO NAS was able to get more than YOLO V8. And then we notice something else. So when species are underrepresented in the data set, YOLO V8 is still able to detect some of those underrepresented species, but YOLO NAS needs a few more examples than YOLO V8 to be able to accurately detect these underrepresented species. And then here on the first SAIAB dataset, we had our biggest gap in performance. We took this as that this dataset provided a unique setting, which YOLO NAS was not able to deal with effectively. When we moved on to the qualitative results, we saw that it is not that YOLO NAS is not able to detect the fish. It is able, but it is a matter of misdetections again. So here you see it detects the BRAV system as a fish. Yeah, so this is consistent with the other results that we saw on the deep fish data set. That's another example of that. And then moving on to the last data set. So here the performance gap was much closer again, but again, YOLO V8 performed best. So it showed consistent performance over all the data sets. So we concluded that YOLO V8 is better suited for the fish detection task than YOLO NAS, as it is robust to different underwater environments and handles complex backgrounds effectively and also handles non-fish objects effectively. However, we do not disregard YOLO NAS as being useless because of the good performance it had on the smaller fish species. And also the problems it had can be fixed by including more negative examples. So if you include more of the BRAV system, it will eventually be able to get it right. So Here's my references. Thank you.
Thank you for that, Kwedi. So I'm gonna take one quick question because we're running out of time. I'll take that.
It's awesome. Thanks, Kenneth. So how do you see the next steps within this? Like, what is the feasibility of using these AI algorithms to automate the browse processing? Where do you think we are, and what's next?
So currently what I'm working on is using synthetic data. So training AI models to create synthetic data sets and then using them as an augmentation to the current data sets that we have. I'm hoping that this will be especially useful when maybe a species doesn't have a lot of examples in the data set, and then we can use the synthetic data to for the model to learn better on that specific species.
Much. Quick few housekeeping rules before we move on to the next speaker. There's a clock behind you there. So as you are presenting here, please keep a look out there and stick to your 8-minute allocated time so that we have the 2 minutes for questions. Also, when you are starting your presentations, please press the space button so that it flows onto the online people as well. And lastly, move closer to the mic as you are talking. Thank you very much. So the next speaker is going to be talking about ocean acidification and climate change, which is Nika Abigail.
Hey, good morning, everyone. So I'm going to be presenting on the impact of ocean warming and acidification on red algae, part of my MSc. So just to give you guys a bit of background on the importance of macroalgal habitat. So they are ecologically and economically significant because a lot of coastal species depend on them as a food source and also for shelter. And they're also vulnerable to global change. So what are some of the expected changes that they will go through? We know as a result of increase in anthropogenic carbon dioxide emissions, there's been an increase in overall temperature, and this affects our ocean ecosystems as well. And then another phenomenon that they will experience is ocean acidification. So basically what this is as a result of the increased carbon dioxide concentration, the ocean carbonate chemistry is also disrupted. So what happens is there's an increase in the dissolved inorganic carbon in the water system, as well as a decrease in the pH. So the significance of this research, as a study species, I've chosen Plocamium corallorhiza. And the reason why we chose this is it's a dominant canopy forming algae in Algoa Bay. It also serves as a food source for economically important abalone. And it's also been shown to provide nursery habitats for juvenile sparrows within the bay. So it's also significant because it can serve as a refuge species. So basically, coastal species that are vulnerable to ocean acidification, for example, calcifying species, they can be negatively impacted by the abiotic changes that are occurring. And macroalgae, because they are photosynthetic, what they do is they utilize the dissolved inorganic carbon within the water system. And they can actually alter the changes and actually serve as a refuge for these other species that are occupying the ecosystem. Another significant thing about this research project is it's a multi-stressor study. So we're not just looking at ocean acidification, we're also looking at temperature and also an interacting effect of acidification and temperature. And it's important to do studies like this because within ecosystems itself, organisms, they don't just have one factor influencing them. They have multiple abiotic factors influencing them at the same time. So the aims and objectives. So the broad aim of the study is to assess the potential impact of warming and acidification on Leucemium collaterata. The objectives, we're going to measure the physiological response as well as the biochemical stress response of these organisms under the different conditions. And as an hypothesis, based on similar studies, we can expect a negative response under the temperature conditions, and the increase in carbon dioxide availability under acidification treatments might actually benefit these organisms. And under the interacting scenario, the increased CO2 availability may actually alleviate some of the temperature stress, but that's actually what we want to investigate. So as a study area, we will be collecting specimens from flat rocks within Algoa Bay. The reason why we chose this area, as you can see in the picture, there's a lot of rock pools there which are dominated by Leucemium species. And the treatments for our experimental setup are also based off of monitoring studies done within this area. So for methodology, we're going to collect specimens from the field at flat rocks, transfer them to the lab, house them in experimental microcosms with different temperature, pH, and a combined treatment. And then after the lab work is complete, we're going to analyze them for indicators of health. And then you can see in the picture, the pH is going to be controlled using pH computers. And then we're going to control the temperatures using normal aquarium heaters. So the post-treatment analysis, we're going to look at physiological indicators of health. So we're going to do measurements of growth and then also photosynthetic indicators. So we have that device there called a diving cam. So it's basically, it can be used, it uses fluorometry, and it can be used to measure the photosynthetic yield and then also different indicators for the chlorophyll levels in the plant. And we're also going to look at biomarkers of health. And basically what this is just going to do is going to see if these plants are eliciting a stress response to the different treatments. And the biochemical analysis can be done using a spectrophotometer. So my research timeline, basically, I'm just finishing off my proposal stage now. We've started procurement for the lab. Beginning of next year, once we get back in January, we're going to set up the lab and start off a bit of a pilot study and then begin data collection. So anticipated outcomes, I'm going to complete my master's thesis. Chapter 1 is going to focus on the physiological stress response. Chapter 2 is going to focus on the biochemical stress response. And then we hope to publish at least one paper in a scientific journal. So what is the significance of this research? It's going to contribute to the knowledge gap in studies in ocean acidification, as well as multi-stressor studies, which are relatively novel research areas. And the data collected from the study can also be used to assist stakeholders in future conservation efforts. So if the response to these changes is often species specific, and if Leucemium responds positively to these global changes and have the potential for refuge potential, basically it can be used to advise on conservation efforts in habitats where these species are dominant. So that's it from me. Thank you. I hope the question.
Thank you very much for sticking to time. We have time for a few questions.
Thanks so much, Abigail. A very interesting plan. Do you have an idea already of which biochemical indicators you're going to be measuring?
Yes. So one of the things that happens with plants when they're under stress, they generally generate reactive oxygen species during photosynthesis, and then they have mechanisms in place to get rid of these bad oxygen species, basically. But when they're under high stress levels, sometimes the scavenging enzymes, they're not enough to get rid of the reactive oxygen species. So basically what we're going to look at is reactive oxygen scavenging enzymes like catalase, superoxide dismutase, ascorbate peroxidase. And then we're also going to look at a indicator of lipid peroxidation. So we're going to see if oxidative damage has happened to the cell. And one of the ways you can do this is by measuring MDA levels in the plant extracts.
Round of applause. Just to change things up a little bit, we're going to have an online talk now from Adam Sluther, and he's going to be talking about imagery and artificial intelligence in the benthic ecosystems.
Is everything good? Can everyone hear me?
Yes,
Everything's good?
Yeah.
Awesome, thank you. Yeah, so thank you for the introduction. My name is Luther Adams. I am a PhD student at Rhodes University. Let me see if I can hide this. Here we go. And I'll be presenting a bit about my PhD, but firstly, I would like to thank all my supervisors for collecting this data over the past decade, and also SAIAB and all the other partners that have assisted in collecting this data. So as I said, over the past decade, SAIAB has been collecting roughly 100 ROV and BRAVs, and this is a lot of data with very few people to actually annotate and process the data. And this data is being collected between Mossel Bay and Sodwana Bay, on the continental shelf between 30 meters and about 300 meters deep. So the aim of my thesis is to investigate and map the benthic invertebrate communities on the eastern continental shelf of South Africa, and to also identify the essential fish habitats of 74 using AI. So as I said, the first part of my PhD is this computer vision component where I will attempt to collect abundance data that will then feed into the second component of my PhD, which is to map the benthic invertebrate communities. From that, I can then use some BRAV data and the ecosystem map that I've generated to identify the essential fish habitat. And then to build on that, I will then further develop monitoring protocols of ROV and BRAV, because, for example, ROV in-field sampling techniques haven't been as standardized as BRAVs have been. So the issue that we have with biodiversity data is that we have this long tail where we have incredibly common species and we also have incredibly rare species. And computer vision models do not like this unbalanced data set. The other issue is that more often than not, computer vision models generalize very poorly, meaning that if you train a model in Algoa Bay, for example, as soon as you try to deploy this model in Cape St. Francis or East London on the same organism and in the same habitat, it generally just doesn't perform as well. And then, like Kenneth has also mentioned, is that collecting and developing this training data needed is expensive, both in human resources and then in time. So the aim of this chapter is to explore and evaluate computer vision to detect and quantify benthic invertebrates. I will do it for some fish, but for the stalkers, mainly benthic invertebrates from ROV data. And I'll do this actually using a two-step process. So in the first step, I found this amazing paper that basically splits this task into two steps. So first object detection and then image classification. And the main thing that I want you to remember here is that all I want to do is to detect organisms, count them, and identify them with high fidelity. So the computer vision model that I'll be using, as you've heard before, is the YOLO version 8 model. And I will basically train this on different habitat types of labeled incredible amount of images with lots of annotations. And I will only actually be using the class invertebrates. So all I want to do is detect all the invertebrates in all the frames, so everything other than fish that we see on the ROV. And then the second step is the classification step, which uses this amazing computer vision model, which is actually a multi-model, and it uses the advancements in natural language, large language models like ChatGPT, and then computer vision to sort this out. And basically what it does, it pairs texts with images where it actually uses the words that you assign to an annotation in figuring this out. So how it basically does is, I want you to imagine in multidimensional space, you've got 74 in carpenter, 2 silverfish, somewhat similar. One has a big dot on the side, and then another fish, a gurnard, it's red. And in multidimensional space, the computer vision model sees all of these as fish, and more similar looking fish will be related closer together. But what it also can use now with this, because biodiversity data has this rich text information that we have, basically the tree of life with it, we can now basically separate these clusters out better by using natural language. And you can add anything. You can add species names, common names, you can add color, form, shape to it, and it's basically better. It basically understands the images much better than conventional computer vision models. So what I've done is I have used all the detections that I took from the invertebrate classification model, and I have then I used it to train this CLIP model, which basically just sorts all these detections, all the species into different buckets. So here are some results. So I've trained this object detection model to about 69% accuracy, but I've actually managed to improve it earlier this week to 89% accuracy. And as you can see, it does a fairly good job at disaggregating the invertebrates from the background, but it also has some problems of pseudo replication where it has these false positives, or it has, or detects a single organism twice. And then in the CLIP model, I've trained to about 90% accuracy. And as you can see, this is a sea fan, Astromyricia fusca, in many different settings, and it was able to cluster all of them together. So as you can see, what I've been able to do is to detect organisms counted to a reasonable level, and then identify it fairly well, which I was incredibly surprised by. Now I want to try to also to move this over to browser, use the same pipeline, and also use the same model findings and apply it to browse data and see how well it performs. But this can also be used for fish or for any method that uses cameras to detect, count, or monitor organisms in the wild. So the way forward for me is to further develop these models, and I'll be doing it like with 74, as you can see in the video there. And then using all that abundance data from these models, I can then map these ecosystems. Then I'll measure some 74, and then hopefully I can identify and map the essential fish habitats of 74 on the shelf. And yeah, thank you for listening.
Thank you very much. We're gonna take a few questions. Sure.
Thank you, Luther. I really like your graphics. I'm acquainted to your type of presentations, and I really like it. One question, especially in relation to the browse, could you if a fish, say fish A, goes back and forth in front of the camera, can you recognize it to that level? Do you count it again, or what do you do? Thanks.
So thank you very much for your question. So for something like that, if it's extremely difficult and challenging for you to do it as a person, it will most likely be just as challenging to do it with a computer vision model. However, there are models that are able to basically track several frames and remember what it saw. So if an organism or fish, whatever, is extremely distinct and you can identify the individual, which is difficult or maybe impossible, then yes, it can do it. But for fish that all of them look the same, unfortunately not.
Thanks, Luther. That was really cool. I have a question about the CLIP model. Can you, it sounds really promising. It's a nice way to integrate all that additional information that we know about each species. Can you incorporate like narrative type text into, so like you've got all these guidebooks with species descriptions and descriptions how you can identify the species. Can you plug in that like kind of a paragraph of information about that species, and would it work in that way, or does it have to be kind of how the classification would work? So family, genus, species type
No,
thing?
you could, thank you very much for the question. You could use any type of natural language. So you could then have that description for that species, add it to the classification. So I don't think there's any limit to the length of text that you can add per annotation. Then you could also create a search engine for it. So if you say, I want the yellow fish with the blue stripe above the pectoral fin, and it should be able to then find all these different clusters of things, and then you can choose from there. It is, yeah, it's just amazing. And I only discovered this model like after I did my proposal earlier this year. Sorry, I can't hear at all.
Sorry, my fault. Does it work both ways? So can you have an image and can that get then turned into narrative text? So for example, if you've got really nice images of juveniles of different species and adults, etc., can you have them turned into narrative text that can then be fed into the model? Is there such a thing?
Yes, and yes, and I have tried it, but what I find is that more often than not, these open models that can do exactly that, they have never seen our type of data. So they've seen fish out of water, and the text that basically can auto-generate from that is a man holding a fish standing on a lawn next to a specific estuary or something. But because our data is like, we are the holders and custodians of this data, and more often than not, it's not in the public domain. And most of these models get trained on public data. It just doesn't perform as well.
Thank you, Luther. A round of applause again for Luther.
Thank you.
We're going back in-house now with Ms. Cweba Siwe on seascaping in the Algoa Bay.
Morning, everyone. Today I'll be presenting my master's presentation on seascape mapping and modeling shallow water habitat mapping around Cape Recife. So first off, seascape ecology is defined as an emerging science that takes techniques and concepts from landscape ecology and applies it to the marine environment. This enables us to describe the effect of seascape structure on biological assemblages. So for example, area, type, quality, and spatial arrangement of patches in a habitat will influence the abundance, distribution, and life strategies of certain organisms. So when we look at seascape structure, this structure encompasses the composition and configuration of patches in a habitat. Now, when we talk about composition, we're looking at the number of patches, patch size, and type. And then when we look at configuration, we're looking at the spatial arrangement of such patches in habitat. So we also look at the concept of habitat heterogeneity, which refers to the number of different habitats in a certain area. When you have different types of habitats in a certain area, this will influence the species diversity that you may find. In benthic environments are usually considered to be heterogeneous because it has, in a given area, we have different patches of different types of habitats. So the benthic habitat plays a crucial role in the marine ecosystem. For example, sessile invertebrates and macroalgae provide food and shelter to certain fish species and other invertebrates. However, there's many common threats that affect this habitat, such as climate change, trawling, mining, coastal development, and shipping. So the effect of seascape structure on biological assemblages can be determined using habitat mapping. So effective mapping of the seascape is crucial because it will help us form a greater understanding of the seascape as a whole and how this will affect the composition and distribution of biological assemblages. It will also give us a visual representation of the seascape depicting its structure and the boundaries present in the habitat. Through mapping, we can also form baseline spatial data for marine spatial planning, monitoring, and conservation efforts. So in my project, while we were reading, we saw that there's little information available on the habitat structure of the reef around Cape Recife. So this study will aim to combine and interpret multi-beam data with drop camera images in order to map and classify the benthic habitat at the western corner of Agoa Bay. And then our objectives would be to classify substratum type and the biological components of the benthic environment using the images. And then after that, we will classify and map the benthic habitat based on seabed characteristics, bathymetry, and biota using multi-beam and drop camera images. So my study site is Cape Recife, which is a prominent cape on the southern point of Agoa Bay. It is influenced by the Agulhas Current, and it's made up of sandstone rock platforms encircled with pools and gullies. So the methods we used to satisfy objective 1 was underwater drop camera sampling, and then we took the images we got from the sampling. We identified the substratum type and the biota found in the image on CPCE, and we used Katami as a classification naming standard. After that, we undertook a multivariate analysis on primer. So these are the preliminary results from my study. For figure 4, we have substratum percentage cover. And what I saw that almost 57% of my sites were dominated by rock. And then we have the other sites that that give us more information is that B2, B22 and B34 were mostly fine sand. And then for figure 5, which is the biotic percentage cover, what I saw is that 31% of my site had sessile invertebrates, and then 26% had folious macroalgae dominant. So after we did our statistical analysis, what I saw is that for all our biota, which was the encrusting microalgae, folious macroalgae, and sessile invertebrates, there was a higher percentage cover where there was rock. So my discussion is, where there is rock present, there is a large macroalgal and sessile invertebrate percentage cover. And then where we have sites that are exclusively sand, we have little to no macroalgae or sessile invertebrates. So I hypothesized that the substratum type influences the biological assemblages observed at each site. And with this knowledge, we'll be able to predict the biological assemblages based on the substratum type we may find at that place. Thank you.
Thank you very much, Ms. Weber. Any questions? Going once, going twice. Sold.
Were the measurements only done in the subtidal areas, or did you actually go into the intertidal zone and map the different habitat types in the intertidal area? Because there'd be big differences between the two, and in terms of availability as well to organisms in the intertidal area versus the subtidal area.
Offshore than the intertidal area, if I'm not mistaken. So mainly because we also have multi-beam data, so the vessel won't be able to, the second objective is using multi-beam data. We're gonna take our images and ground truth the multi-beam. So I think we moved a bit offshore.
Oh, thank you, Mr. Weber. A round of applause again. And we're going back online now, and we're diving into diversity with Ms. Nicole, sorry. Are you there?
Yes, hi, I'm here. Can you hear me?
Yes, go ahead, Nicole.
Okay, perfect. Let me share my presentation. Okay, can you see my screen?
Yes, go ahead. Perfect.
Okay, perfect. Hi, everyone. As mentioned, I'm Nicole, and my supervisors for this topic is Professors Nico Smith, Victor Wepenaar, Luke Brendonk, Assistant Prof. Ellie Thor, and Dr. Marlies Struter. So I'm not going to reiterate the title. Let's just dive right in. So I just want to minimize this. So just a little bit of background. The Northobranchia species are also known as annual killifishes, and they have become increasingly important models in biomedical and ecological research. So these killifish are a keystone species that inhabit annual desiccating savanna pools, also known as temporary pools. And due to the seasonal nature, the killifish have developed a rapid life cycle. So with the life cycle, at the beginning of the rainy season, the pools fill up with rainwater, stimulating their desiccation-resistant eggs to hatch. Their larvae then grows rapidly, reaching sexual maturity, allowing them to breed. The adult fish only live a few months, and all the adults die when the pools dry up, but their eggs are able to survive this dry period within the sediment. This life cycle of theirs also affects other ecological processes, such as parasitism. As pools can only contain a single generation, Thus, this can result in the host availability fluctuations, influencing the timing of interactions between the parasite vectors and the fish. So metazoan parasites have been previously studied once in 2017. And since it is known that the Northobranchius killifish are exposed to various parasites in their natural habitats, little is known about the parasitic communities in the temporary pools, as well as the role that they play in transmitting parasites to their final host. For the study, we sampled in Karangani Game Reserve in southern Mozambique. And just to magnify our sampling area, this is an overview of the 5 sites in which killifish were found and sampled. So at the 5 sites, the killifish was sampled with sweep nets, then placed into plastic bags and transported back to the field lab, where fish from the same pool were placed together in aerated containers. Prior to dissection, the 59 fish were euthanized via double pithing, and then a photo and fin clip voucher was taken for morphological as well as genetic analysis. So for the parasite assessment, the fish were examined externally and then internally, where the eyes, the gills and operculum, as well as the body cavity and internal organs were screened for digenians. Any digenians were present were counted and preserved following standard protocols. And these parasite counts is what we use to determine the parasitic prevalence as well as the infection intensity. So for the results with the external assessment, encysted Metasaccariae were located on the dorsal, caudal, as well as anal and pectoral fins of the fish. And these cysts were removed by brushing through the fins with a fine needle tip. So more encysted Metasaccariae was found within the nose cavity as well as on the tongue, and some of these cysts were excysted on site. For the internal assessment, the gills from the fish were removed, and the gills were screened by brushing through the gill filaments to dislodge any encysted Metasaccaria. And on this slide, we can see there is at least 10 cysts in this one gill. Regarding the cranial cavity, encysted Digenian Metasaccaria was located on the cranial cavity connective tissue, whereas free-moving Digenia were found within the cranial cavity. So once all the cysts were excysted, they were viewed under a microscope with a differential interference contrast, also known as DIC function. And with this, we found 8 morphotypes, with all but number 4 being the excysted Digenia from the killifish. With number 4 in the purple block, it's the image of the freely moving Digenia from the previous slide that was found within the fish's cranial cavity. So for the prevalence and infection intensity with our first site, there was a Digenian parasitic prevalence of 20%, with an infection intensity, going forward, I will abbreviate it as IF, of 38 to 41 for the cysts, and for the freely moving Digenia within the cranial cavity, the IF was 1 to 3. Regarding our second site, there was a parasitic prevalence of 40%, with an IF for the cysts of 1 to 2 and 0 free Digenia moving in the cranial cavity. In our third site, there was a 95% prevalence, with an IF of 1 to 192 cysts and an IF of 0 for the free Digenia. In our fourth site, there was a parasitic prevalence of 90% for the Digenia, with an IF of 1 to 46 for the cysts and 0 for the freely moving Digenia. And for our last site, there was a 57% parasitic prevalence of Digenia, with an IF of 1 to 9 for the cysts and 6 to 14 of the free Digenia. So overall, the Digenian parasitic prevalence for all of the sites was 78%, with an IF for the encysted Digenia Metasaccaria being 1 to 192 for the infection intensity and 1 to 14 for the free Digenia. So preliminary molecular data indicates that there were 7 genotypes as shown on the slide, and they range from Heteroorchis criminifer, the Diplostomidae, post-host Diplostomum species, the Echinochasmidae, the Microphalloidae species, as well as the Diplostomidae CF Diplostomum. So there's only 6 on the slide, but with the next slide, I'll introduce you to the Paraharmotrima caranganiense. And this is the last genotype, and a metasacarial stage was found in one of the killifish from our 4th site. And interestingly and excitingly, the adult stage of this parasite has been previously described in 3 African terrapins in 2022. This allows for the completion of this parasite's life cycle, which will be presented at another conference. So just to compare the study to the one that was previously conducted in 2017, so the location of the parasites in or on the host from the previous one was located in the muscle, the cerebral cavity, the gills, and abdominal cavity, whereas with this study, it was located in the muscle, cerebral cavity, the gills as well, along with the fins and the tongue. Regarding the parasitic prevalence percentage and the infection intensity within the cranial cavity, comparing their species of Apotemon and the Apotemon species in their fish's cranial cavity, along with our Tuladelphus-like, their prevalence was 9% with an IF of 3 to 9, whereas we had a 6% prevalence and a 1 to 14 infection intensity. Thus, their prevalence was higher, but our infection intensity was higher in the study. Regarding the prevalence percentage of the metasacarial cysts and the infection intensity, they had a prevalence of 30 to 100% with an IF of 1 to 807. And we had a 78% overall parasitic prevalence with 1 to 192 metasacarial cysts. So they had a higher prevalence as well as a higher infection intensity. Regarding the morphological characterization, they did not conduct any morphological characterization, and this study has, and both studies have both done genetic characterization. So this study is the 2nd study to date, and this information can enhance our knowledge on the natural exposure of killifish to parasites in temporary pools, providing information on the parasite diversity and their distribution. With the one parasite that has an adult described in African terrapins, this explains the potential role that the killifish or the Northobranchia species can play in the parasite transmission. Thank you.
Thank you very much, Sontobi Doctor. I'll just take one quick question and then we'll break. Can it take us?
Thanks so much, Nicole. First of all, are you parasitologists going on a pronunciation course on the scientific names? They are really extensively long. Second, I just wanted to, as curiosity, I wanted to ask you if maybe I might have missed something at the beginning. Do these parasites get established on juvenile or youngest life stage? And if so, what do you think it could be in terms of potential threats to the population from an early stage point of view? Thank you.
So with the killifish and the parasites within these pools, so they do parasitize them at a young stage, so from the larval stage, not from from when they're eggs. And the impact, if I remember your question correctly, or the life threats that it can have, is that just based on the previous study in which they found the Apotemon species, the one that was done in 2017, this parasite can actually alter the behavior of fish, making them more susceptible to predation. So that's a life threat that that specific parasite has on that fish. It actually makes them more visible to predators. So they conducted a study in which the fish actually jumped out of the water when they were infected with this parasite in their brain. So yes, these parasites can actually have the potential to alter the fish's behavior. So I would say, if that answers your question.
Thank you very much. So in the interest of time, unfortunately, we are going to have to continue with the next speaker. We will be going to Mr. Dube Tembelihle. Is it Mr.? Mrs. Dube, which she's in-house, and she'll be talking about designing effective long-term monitoring programs. So a quick round of applause as she comes to the stage.
Greetings, everyone. As introduced, my name is Tembelile Dube. Okay, so I'll just dive straight into it. Utugela Banks is a marine protected area located in the KwaZulu-Natal province between Richards Bay and Devon. Both commercial and recreational line fishing is known to be taking place in the mesophotic reefs of this MPA, which are historically important fishing sites, and therefore long-term monitoring is very important or essential in order to manage impacted fish diversity in this MPA. So for many agencies, they lack financial support, and therefore managing the MPAs as a massive challenge for them. And this is how then my study fits in, as it was aimed at designing an effective long-term monitoring program for the Utugela Banks. And the objectives were to identify environmental as well as spatial drivers of demersal fish assemblages, and also to conduct a baseline assessment of fish populations in the MPA management zones. The third one was to determine the optimal sampling requirements for an LTM program. So for our methods, key reef fish systems were identified based on existing maps and also mapped using multi-beam echo sounder. Following that, we classified reef systems according to their relief, whether they were high or low relief profile, and also identified random sampling sites using GIS. Following that, we conducted surveys, stereograph surveys, and this technique was selected because it's a robust and also non-extractive tool that is cost-effective as well. So data that was obtained was annotated using software called Event Measure software, where data on abundance as well as size of the fish was obtained. Following that, the statistical analysis were conducted in RStudio as well as Primer, where analysis such as NMDS, CAP, Pemanova were conducted, and also INEX for species accumulation caps. So our results showed that the combination of community analysis, it showed that both depth and habitat played a significant role in the distribution of fish assemblages, and this highlights their role in structuring reef fish assemblages, which then helps us to stratify samples in the long-term monitoring program. And also the deeper sites or the deeper reefs had high variability compared to the shallow reefs, which means that deeper reefs will have to be avoided for the Utugela LTM program and only focus on comparable depths. We also saw the differences between the northern reefs as well as the southern reefs, where they showed different assemblages, and also the differences between the southern control zone from the northern control zone. So the cross-validation from the CAP showed that the overall success was 71%, where both Umloti and Zinihat Edge showed 80%, whereas Tapenza showed the smallest percentage, which was 50% accuracy. And then the species accumulation curves that were connected in order to determine whether we had sampled a representative diversity, and the MPA, they showed high variability in sampling coverage among the different zones of the MPA. And this was due to poor logistical constraints, such as poor sea conditions that led to unbalanced sampling within the different zones of the MPA. And then the species accumulation curves show that we'll need about 8 to 38 samples in order to detect 90% change of the available species within the MPA. And then the power analysis show that 36 samples are sufficient in order to detect a 10% change in slinger abundance at the 0.05 significance level with a statistical power of 80%. And the reason why slinger was selected, it's because it was the most abundant species in the Hotugela dataset, and also it's one of the important species in the recreational and line fishing. So using the data that we obtained from the Hotugela dataset, we then proposed a monitoring schedule, the Hotugela banks, which is managed by the Ezemvelo Wildlife. So they could consider monitoring Hotugela banks where they identify one reef system per management zone and also one northern as well as southern reef, which will then amount to 6 reef systems within the MPA. And also they should avoid deeper reefs, but specifically for this Hotugela LTM program, and focus on comparable depths. We also suggest that they should collect 36 samples per reef monitoring cycle, which will then amount to 216 stereograph samples. And this could be done in a 16-month process where 1 month data is collected in the field, for 12 months video annotation, as well as data management. And then for 3 months where reporting is done. So this could be repeated on a regular basis. And then the Ezemvelo KZN Wildlife should also consider the entire MPAs, like the MPA network, for instance, where they could survey Hotugela banks together with Alwal Shoal, followed by Smangaliso and Protea banks, followed by a two-year break of video processing, as well as report writing. And these could be repeated every 5th year. Thank you.
Thank you, Mr. Mbelihle. We're going to take 2 questions. I don't know if I missed it, but what was the difference between the depth between your shallow and your deep reefs? So what were you classifying as a deep reef and a shallow reef? What depth were your deep reefs versus your shallow reefs?
Oh, okay. So for the shallow reefs, they were ranging between 8 meters to around 90 meters. So basically, because we were focusing on mesophotic reefs, so the mesophotic reefs ranging between like 5 meters to around 100 meters, and then the reefs past 100 meters were considered as deeper reefs.
Thanks. I think it's great to see that your science can be put into practice or into policy. And I was wondering how Ezemvelo Wildlife has have you presented this proposal to them of changing their monitoring or how they could potentially improve their monitoring, and have they taken it up?
It hasn't been presented yet, but the plan is to present it to them after finalizing everything.
Thank you very much. The next speaker back online again is Miss Leroux, and she's going to be talking about the hidden world of tripo. Teach me a word.
Hello, it's Trypanosomes. Can everyone hear?
Oh, what you?
Let me just share my Okay.
Yebo.
Okay, thank you. Thank you so much for the introduction. I am so excited to present to you today the hidden world of trypanosomes and a dual approach to African freshwater fish. So just a quick overview, because I will just present on a part of my master's degree. So my master's degree focused on marine and freshwater fish trypanosomes to describe new species and to potentially identify new hosts. So just a recap of 2023, I described 3 new trypanosome species in marine fish, and that was submitted to the Parasitology Journal for publication. So for the next part is the freshwater fish trypanosomes in Southern Africa. So just a quick introduction, trypanosomes is a genus of Cunitoplast, which is a unicellular parasitic flagellate protist. These parasites can transmit through different hosts and using a vector. Trypanosomes is a very broad range, including fish, reptiles, mammals, and amphibians. And like I just said, they require an invertebrate host to infect fish. They have a very worldwide distribution, and they are known to cause sleeping sickness in human and mammals. South Africa is one of the most biodiverse countries in the world, but only 3 trypanosome species is known at this stage. And later, Baker actually suggested that all of these species can be synonymized to Trypanosome leucoside, having 2 little differences in morphology. So a better description and understanding of these parasites can help to determine the ranges and vectors of distribution and the effect it has on biodiversity conservation, management of fisheries and agriculture, and fish diseases. Why leeches? Vectors is an organism that acts as an intermediate host for these parasites. Some vectors are able to move far distances, and leeches is known to transmit this pathogen through feeding from fish hosts. Therefore, leeches can act as a vector. So my basic hypothesis is the current freshwater fish trypanosomes and diversity in Southern Africa reflects the lack of research rather than the true diversity. So my aim of the study is to increase our knowledge on the diversity of freshwater fish trypanosomes in Southern Africa through morphological description as well as molecular characterization of these trypanosomes. And in order to do that, blood must be collected from different fish species. All blood needs to be screened for trypanosomes, do a complete morphological, morphometical description of all the trypanosomes found, and molecular characterize all of these parasites. So the sample locations was Zambezi River, Tanin Dam, which is part of the Letaba River in Limpopo, and then the Vaal River, Riet River, and Garib Dam, which is all part of the Orange River. Blood of fishes was collected through hand-on from a boat, gill nets, cast nets, electrofishing, and sun nets. So for the morphology analysis, blood was drawn from a fish, stained with methanol and fixed with methanol, and then stained with Ginza stain. And then a drop of blood was placed on a slide covered with a cover slip to screen the live movement of these trypanosomes, and the rest of the blood was placed in a 96% molecular grade ethanol for genetics. It is very important to make sure that your blood smear is nice, thin, and evenly spread. So, all trypanosomes found were photographed and measured, and the following measurements are important to describe. It's the nuclear length, nuclear width, body width, mid-nucleus to posterior end, mid-nucleus to anterior end, the total length, and the flagellum length. So for the molecular analyzers, all individuals that was identified as positive for trypanosomes during screening, genomic DNA was extracted, and 18S rRNA trypanosome-specific primers was used for PCR. A 1% agarose gel electrophoresis was produced to visualize the amplicons under the UV light, and the PCR product was then sent to Inquava for sequencing. Resulting sequences was glassed and then compared with previous sequences to conduct the phylogenetic tree. So for the results, 89 fish individuals were collected and screened from 15 different species. 23 individuals were infected with trypanosomes, which include 2 species, namely the Clarius garipinus and the Oslogranus clatteri. Clarius garipinus had a 90% infection rate, and Oslogranus clatteri had a 40% infection rate. Infected individuals were relatively high infected, with 12 trypanosomes per smear. 2 species were identified. Clarius garipinus were infected with both species, having Trypanosome mucosa, which is already described species, and then the new species, SPD, and Oslogranus clatteri were only infected with the one new species. All the individuals' measurements were placed in primer to construct this cluster graph where the measurements of all the individuals were compared. And as you can see, there weren't significant differences between the measurements, although there were slight differences, emphasizing the importance to do molecular studies to fully describe new species. So for the phylogenetic tree, I use the subgenus Hematomonas, which is the aquatic clade, which includes amphibians, reptiles, marine fish, and freshwater fish. But let's focus on the species or the sequences from this present study. So the purple block at the bottom is all the sequences from the new species, Trypanosome SPD, from the Clarius garipinus and the Oslogranus clatteri, from the Vaal River, Zambezi River, Reed River, and Tenin Dam. And then the top clade is the sequences from Trypanosome mucosa, from the Clarius garipinus, from Tenin Dam, Karoo Dam, and Reed River. So if we look at the bottom clade with the blue dot, this is all the sequences from the Trypanosome mucosa, as I just said, and they all play together with high support, making them a monophyletic clade. And then the top one, which would be the bottom one on the phylogenetic tree with the red dot, is all the sequences from Trypanosoma SPD, which also clade with previously sequenced Trypanosome species. And then on the right, you have a haplotype from all the various locations showing that these species do clade together, although they were sampled at various locations. And this can be due to there's only one freshwater leech described as yet to transmit these trypanosomes. And that could potentially be that this leech do transmit catfish trypanosomes. Yeah, so overall, I successfully described 4 new species and re-described 2 new species of trypanosomes based on morphological features and molecular characteristics. And I added new genetic data to the Trypanosome mucosa clade, and the Tabo River was identified as a new location for these parasites. But further studies need to be done on the behavioral effect of these parasites, as that's not yet known, and more studies on vectors to complete the life cycle, as well using genes like 28S and CO1 to do a more specific analysis for the molecular data. I want to thank you, the following institutions. Thank you.
Thank you, Ms. Leroux. That was really a hidden world from me, at least. So we're gonna take 2 questions. Any takers? Haven't had anyone from up there. Okay. Oh, yes. So.
Why are the trypanosomes so successful as parasites? What are the characteristics that have enabled them to basically parasitize a very wide variety of fish species and also come onto the land as well?
That could be potentially be to the vectors being that successful to transmit these trypanosomes. The vectors, there's a very broad vector range from flies to ticks on terrestrial environments and then the leeches in marine environments. So the vectors having this broad range and being so successful to actually carry this trypanosome make them so successful to transmit to their definitive hosts. I hope that answers your question.
Thanks, Chandra. Very interesting work. I was going to ask you, do you have a link to where there's parasitism by trypanosoma is more successful? Does that size of the systems matter? Does the flow in a system matter, or does it happen mostly in dams or not really connected systems? And if so, if the systems are connected, could you start looking if there is a link to how the potential of how these parasites can spread through along the whole system, the freshwater system? Thanks.
Yeah, so this was actually a difficult thing in the dissertation is we looked at the systems and we have this Orange River and then we have the Letaba River, but they're not connected in a way, and then also the Zambezi River, making it difficult because you can think if the system is connected, then it would be easier for a leech or a fish species to introduce themselves in the system. But now having more than one system, it would definitely be something to look at in the future to see is it the leech that's a catfish specific leech and this leeches occur in all of these systems, or is there something else going on between the systems because they're not connected? No.
Oh, thank you very much, Mr. Leroux. Applause for her. And now we're going back in-house with Nevasamgele. Sitting next to me. Okay, as he talks about the future of D. capensis.
Hello, everyone. I'm Sam Nefa. And I will be talking about the development of ecological niche models to predict the future distribution of Diplodocus capensis in South Africa. So, as we all know, anthropogenic climate change is and will continue to affect both the line fishery and the line fishes within. With regards to line fishes, we are expecting to see declining changes in distribution, changes in growth, and reproductive success. With the line fishery, we're expecting to see declining catches, loss in livelihood, and a general decline in the fishery's ability to contribute to national GDP. Now, this is a big problem in that it calls into question the long-term sustainability of the fishery and our continued benefit from it. But in order to even begin thinking about solving this problem, we must first understand the impacts of climate change on fish populations within the fishery. And how do we do this? This is usually done through looking at things like movement, or we look at long-term distributional shifts, or even shorter-term behavioral movements like migration. We can also look at the physical-chemical impacts of climate change, such as temperature or pH on the physiology of the fish. And finally, we could look at populations and changes in growth, reproduction, and population size parameters. But if we are to do this within the line fishery, we must find a candidate species. And we chose blacktail because the species has been the subject of a wide range of climate change studies that this current study now seeks to complement and add on to. Furthermore, the species is a dominant species within the recreational shore-based fishery in that it supports a lot of livelihoods, making it a priority species for research. And finally, I think most importantly, the species as a spirit, the findings that will be made in the study can be used as analogs for other less available species exploited within the fishery. So this study therefore aims to model the current distribution of Diplodocus capensis and to use physiological thresholds of the species to predict future distribution in response to climate change.
And we did this by running respirometry trials on adult blacktail. And then we will then take these physiological thresholds gathered from those respirometry trials, and we'll use them to constrain models that we want to predict distributions in the current day and in the future. For the physiology, 20 individuals were caught in Port Alfred, and they were transported to the DIF's SAIAB Aquatic Ecophysiology Research Platform, where they were kept in an recirculating aquaculture system. Thanks. So the experimental unit consisted of, the experimental setup consisted of 4 experimental units connected to a filtration system. And each experimental unit had 3 main components. This was the respirometer suspended within a water bath, a recirculation loop within the experimental unit to allow for the oxygen sensor and logger to pick up oxygen concentrations during the experiment, and then a flush pump to introduce oxygenated water in between measurement cycles. And to commence the trial, 4 fish would then be removed from the holding system, placed in the experimental system with 1 fish in each respirometer, and allowed to acclimate for anything between 2 to 3 hours before temperature was then adjusted to the desired experimental temperature. And then following that, the measurement of metabolic rates would then begin. And for the results, we found that with both SMR and MMR, standard and maximum metabolic rate, there was a tendency to increase with increasing temperature. But especially with MMR, we found that there was an increasing sort of variance with temperature with regards to MMR, regardless of whether we temperature or mass corrected the data. Then we proceeded to then go model the relationship of these metabolic rates with temperature using a second order polynomial relationship. And we found that for SMR, there was a positive relationship with temperature that was strong in both for the linear relationship and the polynomial relationship. We also found a similar significance with the MMR, but now the relationship was negative, meaning that as you increase temperature, you are likely to see declining MMR values. We then used these 2 metabolic rates to calculate an aerobic scope, which didn't have a significant linear relationship, but rather a strong polynomial relationship with an overall negative relationship between AS and temperature. This then again tells us that as you increase temperature, you're expecting to see a decline in aerobic scope. But now here we can see a peak at around 16 to 18 degrees, meaning that the aerobic scope is highest at around those temperatures. So after measuring the metabolic rates, we then moved on to measuring the critical oxygen concentration of the fish. And we found that there was no significant relationship. First of all, we wanted to mass correct the critical oxygen concentrations, given the effects of mass and temperature on metabolic rates. So we found that there was no, in this sample population, there was no significant relationship between mass and PO2 crit. And then we proceeded again to use a second order polynomial relationship to model the relationship between PO2 crit and temperature. And we found that the relationship is biphasic. So what happens is it sharply declines until it reaches at about, I think, 18 degrees. And then there it plateaus and starts to increase again. This then, looking at this, we then wanted to plot a hypoxia tolerance index in relation to temperature to see how the fish can tolerate hypoxia. But because of the nature of the relationship, we then had to do a piecewise linear regression to plot that hypoxia tolerance. And this was done so that we can be able to calculate what is called a metabolic index for the fish for incorporating the physiology to the species distribution. So what we found is that from 18 degrees, because this is inverse temperature here, from 18 degrees, from 10 degrees down to 18, there is a sharp decline in hypoxia tolerance. pressed it. So there's a sharp decline in hypoxia tolerance, and which is not as sharp as you then move on from 18 going on to 24 degrees. What this tells us is that the relationship of temperature, it depends on whether it's a cold temperature or warm temperature for the fish. And we found that with the cooler temperatures, the fish tended to have a lesser hypoxia tolerance than they do in the warmer temperatures. And the result of all of this is that what we found is a sort of like a take-home message in all the physiology, that the aerobic scope is biphasic. It tends to top out at like 16 to 18 degrees. And there's a strong link between temperature and PO2 crit, with the relationship also being biphasic, and with cooler temperatures having a greater impact with an optimal temperature of about 18 degrees. And then these physiological indices, and this is now a next step in the study, these physiological indices can then be incorporated into species or ecological niche modeling, whereby we can incorporate them with SSTs, metabolic index, and aerobic scope, along with occurrence, to build a species distribution model. And what we've done for this so far is that we've collected occurrences for the species, around the Southern African coast, which is where their distribution is. And we find an interesting pattern there. There's a break between the eastern and western distributions. And this is interesting because you can see there that that's the coldest part of the coastline. So that's sort of like an interesting find there that we still want to explore as we do the modeling. And we've also been able to then plot the oxygen concentrations around the coast. And we can see that despite there being like the highest point of oxygen concentrations, we still see a break in the species distribution. So which means that the temperature could be the most important predictor of their distributions. So yeah, that's what we want to do at this point. And we are expecting then to have these findings included in our species distribution models, and we can then predict the fish's distributions in the future in response to anthropogenic climate change. This will allow us to have more climate change adaptive management measures. Thank you.
Oh, thank you. Unfortunately, we do not have time for questions. You can catch Mr. Samgelene during tea break. And we're going to move back online. I love this dance. To Botha Murray. Am I pronouncing it correctly? When we're traveling back in time, a century to secure the future.
Thank you so much for this opportunity. Yes, I'm Mary, and we will be traveling back a century to secure the next millennia, redescribing the South African fish ectoparasite Nathia spongicola isopoda nathia collected in 1902. Most research is done through data collection in the form of sampling. However, not all environments are easily accessible. Marine sampling is especially challenging to conduct, as the use of vessels, advanced equipment, and expertise are required when sampling in deeper terrains. Further challenges include the collection of elusive or even extinct specimens. Nevertheless, there are still collections we can consult without the need for current sampling. These collections are found in museums globally. Some of these samples were deposited decades or even a century ago, when a larger expedition occurred or when a species may have been more abundant. Subsequently, the use of museum collections is imperative for updating and continued documentation of species we lack information on. Among these museum collections are marine isopods parasites known as gnathids. They are considered Protilium parasites, as only the juveniles parasitize host organisms such as elasmobranchs and teleost hosts, relying on the consumption of blood, tissue, fluid, lymph, and blood plasma for nourishment. In contrast, the adults are free-living benthic dwellers that mainly focus on reproduction. Furthermore, gnathids are found in shallow intertidal marine regions along the coast, as well as at deep depths up to 300 meters. Gnathids display diverse morphological features between their different life stages, which contributed to much confusion in their initial description. Although some features have been linked between the adults and the juvenile stages, it has still proven difficult to link them solely based on these features. Of the three, males, females, and juveniles, the males have proven to have the most distinct features between the different species, as you may see in the images on the screen, and is why they are predominantly used for species descriptions. The standards for these morphological descriptions were established as early as Menard's publication in 1926 and still undergo refinement to this day. As some of the species predate this standards, it has necessitated the need for redescription to ensure that they conform to current standards. According to Spalding's paper in 2007, there are 12 designated marine biogeographic realms, including the Temperate Southern African Marine Region, or the TSA. Currently, only 9 species have been described from the TSA, of which 4 were described prior to Menard's publication. Both Cicognathia cryptopyes and Gnathia africana have undergone redescription, leaving Gnathia spongicola, first described by Barnard in 1920, to be addressed in this study. Barnard's morphological description was sufficient for his time, limited by available technology that failed to display the finer features. However, he only provided a single illustration and addressed 29 morphological characteristics of Gnathia spongicola. In addition, he identified a second variation, which he named Gnathia spongicola variation minor. Barnard did not include an illustration of the specimen, but instead he listed 8 differences he observed between the two. These differences were attributed to the difference in habitat, as Gnathia spongicola occupied galleries and larger sponges, while variation minor occupied smaller burrows in branching sponges. The hypothesis for the study states what is currently considered a single species, Gnathia spongicola, is instead 2 distinct species. Subsequently, the aim is to resolve the confusion within Gnathia spongicola complex, with the objectives being the redescription of Gnathia spongicola using type material and the morphological identification and description of other species within the Gnathia spongicola complex. So to achieve this, material on loan from the Execo Museum of South Africa was used to redescribe Gnathia spongicola and to clarify whether there were indeed different species. A Nikon light microscope was used to study the features of gnatid type material and were documented through photomicrographs captured by a Nikon camera attached to the microscope. These photomicrographs were of whole gnathids as well as of stained and dissected specimen. The staining was done using lignin pink, which assisted with seeing finer features. And for those that we could not see finer features on the light microscope, we addressed the same, just to give us a bit more comprehensive understanding. So the gnathids were then drawn using a camera lucida, and finally ink drawings were compiled using Adobe Illustrator. Thereafter, all taxonomic information was placed in the descriptive language for taxonomy or Delta program and used to generate a standardized description. So as previously mentioned, Barnard provided a single illustration occupied by limited morphological characteristics. In this study, we considered over 200 morphological characteristics using the Delta program. These included information acquired from a close inspection of the cephalosome or the head, the antennula, the antennae, the mandibles, the pleopod, the pleotalson or the tail, the mouthparts, which include the pilopod and the maxillipede, as well as the 2nd to 6th periopods or the legs. Subsequently, this detailed redescription allowed for a foundation to discern whether Gnathia spongicola variation minor was a paratype of the original or its own species entirely. With a brief glance of both morphotypes, it is understood why Barnard may have labeled both under the same species. However, closer inspection produced additional differences apart from those mentioned by Barnard. We found that the frontal margin of the cephalosome differed greatly between the two morphotypes, with the superior frontolateral processes of Gnathia spongicola being more produced than that of variation minor. Furthermore, Gnathia spongicola had more bulbous eyes, which was 0.4 times the length of the cephalosome, while Gnathia spongicola variation minors were more elongated and nearly stretched the full length of the cephalosome. As observed in the SEM photos, the shape of the mandibles also differed greatly, and the sulcus, found there in the middle, in Nathia spongicola is much deeper and wider than that found in variation minor. These are a few features that indicate a different anterior region of a variation minor, which in addition to Barnard's differences observed, supports the likelihood that they may in fact be two different species. A third morphotype was also found between the museum sample with its own unique set of characteristics. Preliminary observations indicate that it is a completely different species. Nevertheless, it will only be addressed after we have completed the description for the second species. Based on the redescription, it is evident that Nathia spongicola and the additional two morphotypes do not bear striking resemblance to any of the previous described species from the temperate southern African coast, especially in the Nathia genus. However, all three do share the unique feature with Nathia disjuncta, where the sixth perionae separates the fifth perionae and touches the fourth. The study highlights the importance of the use of museum collections in the redescription and in new species descriptions. Although collections over a century ago, it was possible to produce a thorough redescription as well as identify two new species, which aids in the comprehension of our coastal species diversity. By consulting the past, we are now able to understand our marine future a little better. Thank you.
Thank you very much for that nice talk. We have time for 2 questions.
Thanks, Hesmarie. That was really interesting. I was just wondering how the process of, so obviously there's such old specimens, and I was wondering what age and the preservation process, what effect that might have on some of those features that you were using to separate the two types, the minor and the main one, and what your next stages are to maybe, what your next steps are. Like, are you planning on doing some genetic analysis to further pick up if there are differences? Thanks.
Thank you so much for your question. So they are well preserved from the specimen that I have reached. I did not have a lot of each specimen, so we've had a lot more to work with for that of variation minor compared to what we had for Nathia spongicola. I think we were limited to around 9 specimens for Nathia spongicola, and they all shared the similar features that we labeled as the species, or based on the holotype or lectotype rather, that we designated for Nathia spongicola, and they correlate with Barnard's original description. And then when we looked at that, which you labeled variation minor, there were over 160 specimens who all shared more similar features than that compared to Nathia spongicola. And based on what we've seen, I've only labeled a few, because if I were to label all the differences that we are looking at, it would take a lot more than 8 minutes. So we're going on a lot more morphological features. And based on morphology alone, I think we can say we're relatively sure that they're separate species. In terms of the molecular side, it is a bit difficult rather. Some of these species were collected in 1900 and 1902. Some were collected slightly more recently, which was 1961, which is still quite a while ago. And unfortunately, we do not have their preservation records. For the moment, they are stored in 70% ethanol, which I don't think is ideal for molecular. It's already quite a bit of a challenge to sequence gnathids that I recently worked on some that were 20 years old, and they were also in a 70% ethanol, and we had a lot of trouble. So for now, we're not looking at the molecular side, but we are trying to create a foundation where we can reflect on the morphology, understand what we have to work with, and maybe in future are able to then go look on expeditions or so on for these specimens so that we can work towards the molecular understanding of it. I don't know if that answered your question.
I think that was very comprehensive. Thank you. A round of applause. Thank you. And we will go back in house again with Banza Nobuhle as she talks directly to the keynote speaker about the incorporation of indigenous communities for nature-based solutions.
Good morning, everyone. So today I am going to be talking, giving a presentation on my PhD, where I'm looking at inclusion of indigenous communities and finding nature-based solutions for coastal environments. So first, I need to differentiate between natural rocky shores and urban coastal environments. So natural rocky shores are known to be highly complex in structure, meaning that they have those important microhabitats that house a number of different species, and they are known to be biological hotspots because of the high diversity within them. But unfortunately, over the years, we've been seeing more and more of these natural environments being transformed into more homogeneous urban infrastructures through the creation of harbors, marinas, etc. So because of the homogeneity within these urban environments, they unfortunately lack important microhabitats and therefore are known to have less biodiversity in terms of species that occur within them. So over the years, there's been research which has been done by a number of scientists across the globe to try and mitigate the impacts linked to urbanization. So this is called ecological engineering, basically trying to retrofit or modify the artificial infrastructure by creating extra habitats, creating microhabitats that are needed for species. And this is to enhance, of course, the biodiversity within these environments. And there are a number of different approaches that can be called soft approach, which most people refer to as nature-based solutions. There's also the nature-based solutions are basically just replanting of mangrove seagrasses along the coast. And then we have hard ecological engineering, which is retrofitting concrete structures that have different complexity created within them. And then you also have the hybrid, which basically takes a nature-based material and retrofits on the urban infrastructure, which is where my work comes in with EBC. So one thing that we've realized is that across all the different studies that have been done in ecological engineering for urbanized coast, what's lacking is the inclusion of indigenous knowledge systems. So this is where our work comes in by working with the local indigenous community in co-creation of nature-based solutions for urbanized environments. So the aim of my research is to combine nature-based solutions with indigenous knowledge. Firstly, to look at longevity by co-creating and retrofitting two prototypes, which is Imizi and Salthaber, and then monitor their longevity over time. But I'm also looking at biogenic enhancement, basically looking at the survival and growth of mussels on the nature-based structures that we've deployed. And I'm also looking at biological succession by assessing or monitoring the biodiversity in terms of mobile and sessile biota within these nature-based structures. And these are my sites, which are total of 6 sites. We have 2 natural sites, 2 small harbors, 2 large harbors. And we also have Hamburg, which is a site where we collected some of the material we use to create our nature-based structures. So before my work will take place, we needed, of course, to identify a community to work with. And that was the Geisdamer Trust community in Hamburg. So what was needed was to have a sit down as scientists as well as the local community to just lay down guidelines of what's needed, what are the expectations, and also create a memorandum of understanding between the scientists and the local community. And once we had that understanding, we had to get the material, of course, to create these nature-based structures. The first prototype that we created was called Ibizi, which was created using jute fiber, and this material was commercially obtained. And then the second prototype is called Imizi, and it was created using Cyperus sextilis, which is commonly known in Xhosa as Imizi, and it's economically and culturally very important for local indigenous communities in the Eastern Cape, as it's used for sleeping mats, baskets, etc. So once we had that laid down, sorted, we needed to create the actual prototypes. And then the different prototypes, the first one was Imizi. And then within the prototypes, we had different treatments. We had the control, a flat, and the seeded treatment. And the seeded treatment had extra complexity by seeding penna brown mussels on the actual structure. So we looked at a number of things which I did mention on my objectives. Firstly, we wanted to see between the two prototypes, which of the two prototypes is actually maintaining its structural integrity over time. We also wanted to look at mussel growth and survival on the seeded prototype. And also we wanted to look at the actual biodiversity that the structures are attracting by looking at the mobile stuff, by direct counting mobile biota on the structures, as well as taking images of the structures monthly over time to actually look at the sessile integrity biota, and then using those images to estimate percentage cover of the sessile biota. And this is just some of the statistical analysis that I did. And then firstly, when we look at the longevity, we found that for SAIAB, the first prototype, firstly, we found there are significant differences between the prototypes, with Imizi having higher or greater longevity compared to Imizi, compared to SAIAB. And then when we looked at SAIAB, we found that SAIAB only lasts for a period of 6 months. And then also within natural sites, which we initially deployed in, all the structures were lost within the first 2 weeks of deployment, which means that when we're deploying our Imizi prototype, we did not deploy in the natural sites because of the losses that were encountered earlier on. And then for Imizi, we found that they only lasted a total of 12 months. And then with the exception of Mocha, which was a very high wave dynamic environment, meaning that our structures were under a lot of stress and we're losing a lot of structures faster compared to the other sites. And then we found that small harbors, which are more sheltered, had higher longevity compared to the larger harbors. Then when we looked at the mussel growth, there were again significant differences between the prototypes, with Imizi having higher growth of mussels in comparison to the SAIAB. And then we use shell morphometrics. I forgot to mention this. We use shell morphometrics to assess the growth of mussels. And then throughout, we did see that Imizi had higher growth of mussels compared to the SAIAB. And then again, when we're looking just within or across the different sites, we found that there are again significant differences between the sites, with PE having higher growth. And then, so I just showed heights alone, but the combination of height, length, and width for mussel growth was consistent throughout for PE. So PE was one site which had higher growth of mussels. And then we looked at the actual diversity within the nature-based structures. We did find, again, significant differences between the two prototypes, with Imizi having higher species diversity over SAIAB. And then when we looked at within the treatment, we saw that, again, it was the same case with controls and seeded for Imizi having higher, with controls having higher species diversity. And then same thing with SAIAB. Then we look at richness. We found that our controls and the seeded treatment were equal in terms of richness for Imizi prototype, but for SAIAB, only the controls had higher richness compared to the different treatment, which was a flat and seeded. So firstly, in terms of longevity, of course, for Imizi, as I've already said, the longevity is greater compared to SAIAB, and this could be potentially because of attachment stability. For Imizi, they were more stable, meaning that they could nicely attach on the concretes or on the substrate, whereas for SAIAB, it was the other way around. And then for biogenic enhancement, again, because of the stability of the actual structure of Imizi, mussels were able to attach and stay there longer. And then in terms of their growth in PE, having higher growth compared to all the other sizes could be linked to nutrient enrichments that are coming into the Port of Elizabeth. And then when we're looking at biological enhancement, mobile species, because for the diversity indexes, I only looked at mobile species for now, which I'm currently working on the sessile side of things. So we did find that mobile species are higher on the Imizi prototype, as well as the controls too. But then One thing that so far we can say is that IMIZI is the best candidates for coastal rehabilitation, looking at just the two components, which is longevity and biogenic enhancement. But there still needs to be a lot of work that needs to be done for us to actually confirm that in terms of succession, they're also being as effective compared to the soil saver. So we still need to actually do succession, biological succession of sessile in order to confirm and tick all the boxes saying it's good for longevity, it's good for muscle growth, as well as it's good for succession. So yeah, thank you.
Thank you. Unfortunately, we don't have time for questions this time around. It's a very interesting project, so please try to find her during tea time and have that conversation. The last speaker for this session is going to be Makeng Ngulizi, and I think she's online. She's going to be talking about urban protected areas.
Good day, ladies and gentlemen. Am I audible?
Yeah, Bo.
Okay, good. Good day once again. Thank you for granting me this opportunity to present to you the progress I've made thus far in my master's project, which is titled Assessing the Conservation Value of South Africa's Urban Protected and Conservation Areas. This project is supervised by Dr. Fortunate and co-supervised by Prof. Louis. Next, please. When we think of conservation, we often picture the majestic lions or the mighty elephants. But what about the small, resilient, and often overlooked frogs? Frogs may not roar, but they signal the health of our ecosystems. And without these species, the food chain and the ecosystem as a whole will drastically decline. Hence, we have protected areas. Urban protected and conservation areas are designed to protect the biodiversity and the ecosystem services in cities, but their effectiveness for amphibians conservation remains unclear. Amphibians, particularly frogs, are currently facing a global crisis, with 41% of them facing threatened with extinction, which is the highest proportion of all vertebrates combined. Globally, only 22% of amphibian species and 36% of their genetic diversity are protected in designated areas. In urban environments, threats like habitat destruction, pollution, invasive species, and climate change are intensified by urbanization. This study therefore focuses on frogs, which is the most diverse amphibian group in the whole of South Africa, to evaluate their representation within the UPCAs in Johannesburg, Durban, and Cape Town. Next. The aim of the study is to assess the conservation value of South Africa's UPCAs for frogs. This will be achieved by mapping the frog species distribution to evaluate their representation within the UPCAs, to survey the species in the areas with low representation, to assess the quality of the frog habitats in these UPCAs, and lastly, to provide amphibian conservation recommendations based on the results that will be obtained from the study. Next. For the methodology of GIS, the shapefiles that we utilized in the study were obtained from various data sources. So from the Department of Forestry, Fisheries and the Environment, we obtained the support and the SACAD shapefiles. And for the frog records, we obtained them from ADU, FrogMap, and GBIF. These were then overlaid using ArcGIS Pro software. Thereafter, the frog distribution maps were created so as to show how the frogs are distributed within the different UPCAs boundaries. Finally, the percentage of the species representation to identify the species gaps and to understand the extent of the frog distribution in the cities was calculated. Next. For the methodology of how the species will be collected, as I've said, that the surveys are aimed at frog species, which have shown to have a low representation within the UPCAs, meaning that we'll visit the UPCAs show a low representation. Visual sampling will be collected. The collected frogs will be placed inside a jar with little water. The jar will be labeled using a unique field number. The frogs will then be transported to a field lab whereby they'll be identified using their external characteristics to species level. Blood will be drawn, which will be then used to screen the blood parasites of those frogs. And then also, we are going to confirm their identity using DNA barcoding. For the only 2 species per locality will be vouched, and those that will not be vouched will be returned back to their original site of capture. Next. So the primarily results that we got from this study for show that very few frogs are represented. So as you can see from the map, that Durban had the least frog species distribution with only 1%, followed by Johannesburg, and it was only 2%. However, Cape Town, which is our biodiversity hotspot, had a much higher species distribution at 38%. Next. Cape Town is known for its exceptional biodiversity, primarily due to its location within the Cape Proleastic region, which offers different diversity and habitats, also favors the environment conditions for biodiversity. However, urban centers such as your Johannesburg, Bloemfontein, and Pretoria also are showing increase in amphibian records. Next. So these findings that we got suggest that a sampling bias can be seen. Research shows that Western Cape is usually favored when it comes to sampling of frogs in cities. Next. So we can see that we have noticeable gaps in the frog species representation in our UPCAs across Johannesburg, Durban, town. Next. Johannesburg and Durban show minimal species coverage, while Cape Town has better but still has insufficient protection. Next. So in conclusion, expanding the UPCAs and improving habitat connectivity could contribute to amphibian populations in the urban environments. Conservation efforts must be therefore integrated into planning urban UPCAs the long term of survival for frog species within the cities. Next. So since we have conducted the first objective of our study, the next one will be collecting, visiting the UPCAs which show the low representation of frogs to collect our specimens. And after that, we'll be assessing the quality of the surveyed frog habitats. And lastly, we will try to make urban amphibian conservation planning recommendations based on the results we will have obtained from the study. Next. And that's my last slide. Thank you, everyone. And I would like to thank the following organizations with funding. Thank you.
Thank you very much. She was very quick. So we have two questions to take on the floor. People are tired. Okay, one question.
It appears as though there are different factors influencing the diversity of frogs and their abundance in urban areas, such as the size of the areas under conservation, how long they've been conserved for, what the connectivity is with adjacent areas and that. What is the major factor that you think drives the diversity and abundance of frogs in urban areas and which requires the most attention?
Okay, so I'm just trying to make sure that I got the question correctly. So you'd like to know which factor contributes to the biodiversity conservation within the protected areas?
Yeah, I would have thought size was quite important, because if you have a small area in an urban, yes, the size
of the urban area or of the protected area within the urban area.
Yes, yes.
Oh no, size does matter, but what matters most is what is found within that particular urban area. So, you know, frogs prefer a place that's moist. So we have to have something like a pond or a river or something like that. We have to have trees and stuff like that. However, in urban protected areas, even though we have those things, they are not of good quality. So our rivers are there, but then they are highly polluted. We have invasive species within those urban areas. And then often we see that there's habitat destruction happening every once in a while. So therefore, we do not have a rich amount of frogs happening or occurring within our urban areas. So that's what we are trying to find, or rather research on.
So there's an online question that asks, most affected urban areas are the aquatic systems. And during my stay in Johannesburg, I have seen many frogs dead in the streams because of anthropogenic litter, as they also use those habitats. Do you think there could be a way to ensure that they remain stable within this system, given that human population is increasing in cities, in turn the threats we have on ecosystems?
That is my last objective. So I'll try to make recommendations on how we can try to conserve these frogs within the protected areas. But for now, we have seen that most of these frogs are trying to change in a way. They're trying to adapt. That's the better word to use. They're trying to adapt to these factors that they're faced with in our urban areas. However, by adapting, they are also changing, they themselves. So a frog that was there 20 years ago when the environment was a bit healthier, it's not the same as a frog that will be found today. So it changes. And with the statement that you made about frogs being found dead in a river, we are experiencing that due to the different types of chemicals and so forth within our rivers. So we know that the skin of frogs is permeable, so they use it to breathe, hence they get affected and die when they cannot survive or rather adapt to the crucial environments that they are faced with in our urban cities.
Well, thank you very much for that. And thank you for all the speakers. Can we give a round of applause to everyone in this session? Thank you. So we're going to break for tea now and come back 11:30 sharply so that we can get started. Thank you.
Second session. So in this session is going to be very exciting. We have quite a range of different topics, ranging from insects, fish, and as well as ecological topics. And our first speaker is going to be Hlumelo Manshi, who's going to be talking about mosquitoes and how they're being affected by the changes in the environments. So welcome you to the stage.
Hello everyone, I hope I'm audible. My name is Hlumelo, as mentioned, and I will be telling you on how and I'm going to look at the interactions between mosquito and invasive floating aquatic weeds. So just to kickstart, my project is a mere fraction of a huge international collaboration project that tries to look and understand how mosquitoes are being affected by invasive aquatic weeds, or how these two interact. And what does that mean for the warmer world, and what role do these invasive aquatic weeds play in mosquito proliferation? This is my beautiful team, consists of supervisors. I'm sure majority of you guys already know them, and they are here, some of them, and consists of students. Toris will be presenting next. So for my study, I considered 5 notorious invasive weeds, ranging from water hyacinth, caribou weed, water lettuce, Paris feather, and the red fern. So others may ask, why mosquitoes? As we know, we all hate mosquitoes, but why them? The question is that the climate is changing and we're getting warmer and warmer temperatures, and that might influence both the distribution of the invasive aquatic weeds and mosquitoes. And secondly, mosquitoes are vectors of deadly pathogens, such as the West Nile virus, the chikungunya virus, and malaria. And all of them recordedly caused about 700,000 deaths annually in 2020, and these numbers have been recorded to steadily going up. We know that doesn't feel good, right? Okay. So just on the background on how these invasive aquatic weeds alter the system or the community structures. First of all, you will see an increase in shade. The increase in shade is caused by the dense mat of vegetation created by these invasive aquatic weeds, which prevent light penetration to the system. That results into low water clarity. As that happened, we see a decline in sub-immerged microphytes. That results into habitat homogenization and also reduces the dissolved oxygen. And then all of that process, we will see an increase in temperature. And Torres will be telling you on how predators behave in this situation. And then my question is like, how these factors now influence mosquito communities? So these are just field observation pictures taken by my supervisor when she visited one of the sites that I invested invaded by this weed. So in this frame, we're just seeing mosquitoes resting on the vegetation. And on this second frame, if you see, I'm not sure, it's supposed to be a video, but if you see there's those small things, maggot looking like structures there, those are mosquito larvae in abundance in a site that is fully invaded by water hyacinth. And this is water hyacinth in the field. And this is a cool video. I'm not sure why it's not playing, but yeah, it has, you will see mosquitoes just flying out of that vegetation. The main purpose of or main aim for this study was to investigate the relationship between invasive aquatic weeds and mosquitoes. And we had 3 major objectives. The first objective is the field campaign, where we're going to look at the mosquito community dynamics in the field. The second one is to check how these invasive aquatic weeds alter the thermodynamics of a system. And the third one is to quantify the changes in functional responses of mosquito predators. So let's get to the first objective. The first objective will start next year. Early January, we will be visiting KZN, which is a subtropic region of South Africa, and approximately 30 sites will be visited where they will be invaded and also a non-invaded sites. So in each site that we're going to visit, we're going to take a SAS net, sweep through the vegetation to get the predators representation within those weeds, and then we're going to use a deep cups, which are used to sample mosquito larvae to try and also see the mosquito communities within those vegetations. And lastly, we're going to sample adult mosquitoes using traps from Biogen, and we're going to use sugar water as bait. Then for the second objective, it's set up in Vinek. We have a rearing facility owned by CBC here at Rhodes University, where we have tubs full of these invasive aquatic weeds. What we do in there, we're going to have eye buttons set in different depths above 15 centimeters above vegetation, and again, surface and just 15 centimeters below the surface. And then what we observed so far is that above vegetation, we can see that they followed a similar trend, regardless it's being a salvinia or water hyacinth or azolla. That could be maybe those that have been have a high temperature could be the tunnel recordings, but when it comes to water hyacinths, it gets interesting. But the moral of the story, I want you to focus more on the water surface and the 15 centimeters below the water surface. And if you see those temperatures, they range between 20 and 30, and they are within the recorded optimum temperature for mosquito development, which might suggest these invasive weeds are harboring or creating a suitable environment for mosquito to breed in. And this data was recorded towards the end of the winter, which was still cold in Grahamstown. You can imagine if this water stays like this, even that time. So for the third objective, what I did for the third objective, it's trying to understand the development rate of mosquitoes under different temperature and shade regimes. So if we're looking at that first, this first plot, we're looking at time to pupation. So if we're looking at time to pupation, for the first part where we have no shade versus shade, we can see where there is shade, they tend to reach pupation faster, but more statistics needs to be done to conclude to that. But according to the plot, it looks like that. And if we look where we increase the temperature to 25, we see also that it shifts. Those that are in no shaded systems, they tend to quickly get into pupation, as similar trend observed by those that are at 40% shade. I'm sure you're wondering why are these empty spaces? Those were under 80% shade, and none of those mosquitoes that were put in those containers were able to reach the 4th instar. So they died before they reached pupation. And on time to emergency, meaning that from pupa to adult, again, as we can see that those that are under 40% shade at 20 degrees Celsius, they quickly reach adulthood as compared to those that are at 0% or no shade at all. Again, a similar trend is observed from this and this, where we find that where there's no shade, they quickly get to adulthood. When there's shade, it's a little bit delayed. And again, we still have zeros because those that are under 80% shade never went and reached adulthood. So still to do, I'm planning to run a critical thermal limits for all mosquito larvae levels, mostly especially the Chulex, and again, do the same for the adults. I'm sorry, for the predators. And I'm trying to see if both of them can coexist in the changing system. And what does that mean on mosquito control if these predators cannot withstand the temperatures that the mosquitoes can withstand? And lastly, I'm still currently working and running the functional response experiment, which will tell us at what density and size can these predators feed on the mosquito larvae. And also I'll be using different temperatures and different shading regimes. With that being said, I'd like to thank my supervisors and all the parties that played a role in this huge project. And from me and this mosquito, thank you.
Thank you, Phumelo, for the wonderful talk. Unfortunately, we don't have much time. And whilst we're on that topic of time, please, speakers, do try to look, there's a clock at the back there to just guide you and make sure that you stay on time. Secondly, I'll also like to remind you that once you're done with your talk, just make sure that you click after the last slide so that the next one can be loaded for the incoming speaker. With that, we move on to the next speaker. Also keeping to this theme of mosquitoes, it's Toriso Masa-Lesa, who will be talking about the floating aquatic alien invasives.
Greetings, ladies and gentlemen. I'll be sharing my study with you where we ask the question, do floating aquatic alien invasive plants facilitate mosquito proliferation? Phumelo has already introduced the study species that we'll be working on, which are the water hyacinth, the water lettuce, the Kariba weed, the red water fern, and the parrot's feather. The key traits of this species are that they grow rapidly and they also spread rapidly. They form dense mats on the water surface, which then prevents light from penetrating through the water surface and has implications for oxygen levels in that system. They cause massive ecosystem damage as they alter the habitat complexity and also disrupt the native biodiversity. The question is, how do these plants then affect mosquito population, which has not been greatly studied? They can be a source of food from the flowers that they produce, particularly the water hyacinth, and they can also provide algae, which is a primary food source for larval mosquitoes. They can also offer oviposition sites in their leaves where they store small water, they form small water pockets, and they can also protect larvae from predators in their root system and also their leaves. They can also exclude predators which predate on mosquito larvae, preventing their populations from growing large. The aim of the study was to investigate the potential relationship that exists between invasive aquatic plants and mosquito populations, and the objectives were to determine which aquatic plants attract mosquitoes for oviposition, and this was achieved through a mesocosm experiment. And it was the other objective was to determine if the presence of the invasive aquatic plants influences the predator-prey relationship between the mosquitoes and their predators, the back swimmer, and this was done through a lab-based experiment. For the methods, we set up wash buckets behind the Rhodes University Department of Zoology and Entomology, and then this experiment took 42 days where we sampled every 7 days. We took measurements of the physiochemical parameters and also the nutrient content, and then we collected insect samples, which were then sorted and preserved, then later identified. For the predation experiment, the first part, we checked the larval predation rates, whereby we placed 3 aquatic plants, which were the water hyacinth, the parrot's feather, and the azolla. And we also added a native plant, the common duckweed, as a control, and also had a no plant control. Then we had another experiment where we monitored the predator behavior in the presence of this plant, whereby we divided a fish tank into 3 parts, for both vertically and horizontally, and then 1/3 of that tank was divided with a PVC divider, and then we put the plants in that section to prevent them from moving into the other sections. For the results, we found good aquatic microinvertebrates, but for the study, we only focused on the mosquitoes. So here we can see that the parrot's feather and the water hyacinth performed greatly, whereby they had the most abundances of larval mosquitoes, whereas the giant salvinia and the red water fern did not have the most. And as we can see on the red water fern, that the mosquito larvae that we found, they were almost 0. For the predation experiment, we also took measurements for the light intensity so that we can determine if they really do affect the light penetration. And we found that the water hyacinth and the parrot's feather had the least light intensity passing through, but also the azolla had lower light levels, as all of them, the azolla, water hyacinth, and the parrot's feather were below 50. For the predation, we also noticed that in the treatment that had parrot's feather, the consumption was very low, and also as well as in the treatment that had the water hyacinth. Then when we checked for the predator behavior and where it moved, we can see that there was a significant difference between, treatment difference in the parrot's feather treatment for both the horizontal zone use and also the vertical zone use, where it preferred to spend most of its time in the open water here, and also here it spent most of its time in the surface. So it would be in this quadrant over here. That's where it spent most time. So it shows that it avoided spending most of this time in the vegetated zones. So the outcomes of this study show that the invasive aquatic plants do affect mosquito success rates differently, whereby the water hyacinth and the parrot's feather seem to favor mosquito abundances more than the other treatments, but also they play a role in the predator-prey relationships that are experienced by the predator and the prey. So that we can suggest that the protection that the water hyacinth offers the larval mosquitoes is structural protection, whereas with the parrots feather, we are not entirely sure, but we expect that it is chemical refuge that it offers, and more studies need to be done on this to ascertain it. So this then tells us that aquatic invasive plants do affect or influence mosquito abundances, which can be a cause of public health concern. So we can use this results to, we can use this results to determine which plants need to be controlled more, where to put most of our efforts in getting rid of this invasive aquatic plants, as they cause a lot of damage to the ecosystems, and also they pose threats to the human health. Yeah, thank you.
Thank you, Teresa, for the wonderful talk. We have time for 2 questions.
Thanks, Chair. I just want to actually very good talk. I really enjoyed it. But one thing I wanted to maybe find out from you is with these alien invasive plants, can we safely say that with them they can perhaps lead to the extra limited occurrence of the mosquitoes, since I mean these aquatic plants, we tend to find them spreading all across the country. Like I'm talking particularly with the issue of the mosquitoes that are causing malaria, for instance. I mean, there has been incidents where they are said that they tend to be spreading like into those areas that they've never.
I think they could, because they do seem to favor their success rates, and also they prevent predators from predating on them, and also they can also stop their competitors as they disrupt the native biodiversity of that area.
Thank you. We can move on to the next speaker. So the next talk is a taxonomy study on parasites of the yellowfish, and to give this talk will be Tenelo Masilo, and this is online.
Is my screen showing? Hello?
Yes, looks perfect. Thank you.
Okay. Greetings to you all. I will be talking about the redescription and first molecular characterization of Dactylogyrus spinocereus. This project focuses mainly on addressing the lack of knowledge on how parasite communities change over time. To determine how parasites have responded to human impacts in a system can be used. This discipline monitors the change in a group over time, which can be linked to human-environment interactions. With historical ecology of parasitism, preserved specimens could be mined for useful data. At the South African Institute of Aquatic Biodiversity, there is the National Fish Collection. After going through their database, we found that they have Labiobarbarus mariquensis in their collection with samples spanning 60 years. A few months ago, L. mariquensis were collected and are currently being screened. As previously mentioned last year, sampling of fresh material has occurred as a base point for the comparison over time. Fish individuals were collected from the Letaba River, which is in Tzaneen, Limpopo province. A full parasitological screening of both endo and ectoparasites was performed. The parasite taxa found include Monogenia, Digenean Metasaccharia, Myxozoan Plasmodia, nematodes, bivalve larvae, and copepods as shown. Overall, Overall, 79% of the collected specimens were parasitized with intensities ranging between 1 to 27 for all parasite taxa found. With this presentation, the main focus will be on the gill monogenian called Dactylogerus spinocereus, which falls within Monopistocotylia. These spinocereus have been reported from several cyprinids across Africa, and in South Africa, it has been reported on L. mariquensis. So, what are monogenians? Monogenians are parasitic flatworms that inhabit diverse environments such as marine, brackish, freshwater, and terrestrial settings. Most African species are ectoparasitic, being reported on the gills, fins, and skin, and occasionally in the oral cavity and nostrils, while a few species are endoparasitic. They can be divided into two groups, Monopistocotylia and Polypistocotylia. Monopistocotylia use hook-like organs on their hapters to attach to their hosts, whereas polypistocotylians utilize clamp-like structures for attachment. Monogenians are hermaphroditic, possessing both male and female reproductive organs, and have a single host with a direct life cycle. In light of the information that we now know, I want to update the information available on D. spinocereus and provide novel information on the species. The first objective is to complete a literature review on the host and geographical distribution of D. spinocereus. Secondly, do both morphology and molecular work as part of an integrated approach of characterizing the species. This will be done by sampling fresh material, taking measurements, drawing specimens, and digitally inking them. Finally, molecularly identifying the taxa found using partial 28S rDNA and CO1 gene regions and constructing a phylogenetic tree to compare to other dactylogerids. As part of my literature study, only 7 papers were found on D. spinocereus being parasitic on L. mariquensis. The paper by Papena and Thurston 1963 showed morphology done, but no morphology was done in other publications. These sources were found in Google Scholar, NW Library, Margaret Smith Library through Maditava. The hosts that D. spinocereus are known from in Africa are Labiobarbarus alternalis, Labiobarbarus somereni, and Pteromeus nyanzae, and Labiobarbarus mariquensis. D. spinocereus was originally described by Papena and Thurston from L. alternalis in Kenya as Neodactylogerus spinocereus. After obtaining additional material from E. nyanzae in Kenya and L. somereni in Uganda, they placed it in the genus Dactylogerus. These reports largely identified D. spinocereus based on general morphology and comparison to the original description, which does not accurately show the morphological features that truly define the species. In South Africa, the first report of D. spinocereus was from the gills of L. mariquensis in 1983 by Mashego, which extended its known range across the continent. It was mainly reported from the rivers in the Limpopo province. The male copulatory organ was thought to be the key morphological feature for identification. It is evident that not all MCOs are the same. As seen, the first MCO is from the description made by Papena and Thurston, and this one was drawn during the current day study. We compared it with the MCO from D. Matlopong from a paper by Acosta 2022. We chose to include features from D. Matlopong because D. matlopong and D. spinocereus belong to the Varicohini type. These specimens normally have the same key morphological features for identification. Another feature that can be used for identification is the vagina. This is from D. matlopong, and this is from the present day study. Another key morphological features, Next, we have the haptoral structures. On the left is the one from Papena and Thurston, top right is the one from the present study, and the bottom one is from D. matlopong. The upper one is from D. matlopong, and the one below is the one from the present day study. We can see similarities within the structures which show that they all belong to the Varicohini type. They all have 7 pairs of hooks that are similar in shape and have a protruding round shape on all of them. They also have the ventral and dorsal bar. The ventral bar from Papena and Thurston has a pointier tip than those from D. spinocereus and D. matlopong, which have more rounded tips. The dorsal bar from recent studies is more similar than that from the study by Papena and Thurston. When it comes to phylogeny, no studies in Africa exist that had molecular work done on D. spinocereus. So, dactylogirids from around Africa were used to construct a phylogenetic tree. The tree is divided into two groups, the Varicahini type in pink and Carpathicus type in green, using maximum likelihood and Bayesian inference of the partial 28S rRNA gene. The ML and BI tree topology slightly differ for the Varicahini type only. The newly sequenced taxon is in bold, and the species with the highest molecular divergence to D. spinocereus is D. matlopong with the base pair difference of 0.85. In conclusion, this research provides updated work on morphology and represents what we described as South Africa's D. spinocereus. It also provides the first genetic material on this species and the genus in South Africa. We found out that there is no type material for comparison, so this work could be valuable for future studies to characterize and do integrated reporting such as proper genetics and morphology. And lastly, I hope to obtain CO1 sequences next. I would like to thank the following for funding and assisting in fish collection. Thank you.
Thank you, Sanelo, for the interesting talk. We do have time for just one quick question.
Okay.
Any questions? Okay. I just have a question. Have you gotten to the stage where you figured out the rest of the life cycle, or right now it's still just a description?
Right now it's just the description, morphology, and...
Okay, thank you. A round of applause for Sanelo. So we are now moving on to movement ecology, looking at the mullets by Bantini Zico.
Good day, everyone. So as introduced, I'm going to be talking to you about the movement ecology of mullet species in the Kawi estuary. So I'm going to just try to sum up my whole PhD in 8 minutes, which is pretty easy. Thank you. So when we think about coexisting species, there's this concept of niche partitioning, whereby each organism sort of finds its own niche in order to limit the competition that happens. So for example, they can either do this by partitioning the resources that they use, so they can feed on different resources, or they can use different spaces. But then the challenge is when you have species that feed on the similar things and they're also similar morphologically. So an example is the study species that we're going to be talking about, which is the mullets. The photo that's shown here is 8 mullet species caught from a single net. So these species, they feed on similar things. So we want to try to find out what else drive their coexistence, since they feed on the same type of food. So we want to look at the movement patterns of these species in order to determine their difference in terms of the niche partitioning. So globally and even in South Africa, there hasn't been studies done on the mullet species, given that they are also used for food. They have economic importance and socially important. So I want to identify their habitat use patterns, and also these are key species in terms of their abundance. So they can also be used as environmental indicators, as they can be used for proxies for climate change. So the broader objective was to look at 3 mullet species and to look at their habitat use patterns. And we also wanted to look at how future climate change may impact the way in which these species use their habitats to determine if they're going to persist in future. So We used three methods, acoustic telemetry, to determine the habitat use patterns of three species. Then we selected one species, which we used another method, otolith microchemistry, to determine the lifetime habitat use. And the last objective was to look at the thermotolerance range of one of the mallee species that we selected. And using acoustic telemetry, we were able to determine the habitat use patterns of 34 tagged individuals. So these were consisting of 12 groove mallets, 6,000 and 14 striped mallets. And what we can see, so this is 1 year of monitoring data. So what we identified from this data is that these species were using the different parts of the estuary. So if you look at the top graph, which is the groove mallet, it was predominantly using the mid reaches of the estuary. And when you look at the southern mallet, it was almost using the entire length of the estuary, even though there were few individuals for the southern mallet. And then the striped mallet, so this is 1 year data, it was mostly restricted to the lower parts or the mouth region of the estuary. And moving on, the other method that we used was so acoustic telemetry gave us like the fine scale movement patterns, wanted to look at the habitats, lifetime habitat use patterns. So we used otolith microchemistry, where we extracted otoliths. Then we used a laser ablation machine. So what essentially does, it measures the elements, different trace elements within the otoliths. And this you can use to infer which environment this species has been. And the element that we're considering in this study is strontium, which tends to be positively correlated with salinity. And what we found is out of the otoliths that we extracted, majority of them had a movement pattern that we referred to as type 1, which was started with elevated strontium levels. And then after some time, which is probably after 1 year, they moved down into less saline waters. So that could be representative of them moving from the sea into the estuary. And the other type that we identified was a more resident type, which didn't show any much variation within a restricted range of 0.4 and 0.6. Then there was the last type, which was kind of nomadic, using both the estuary and also the sea. Then, so for the last objective, which we wanted to find out how climate change might impact the habitat use patterns of these species in future. So we used the CTM method. So we measured the CT max and the CT mean of the groove mallet. So we just did for one species. And during the experiments, we were also able to measure the rate of opercular beats as the temperature changed. And what we found out for the groove mallet is that it had a broad thermal range, which was ranging from 5.7 to 36.7 degrees. And the rate of opercular beats, it was positively correlated with the temperature. And also the arrows also indicate parts of our graph where the graph was not so linear. So we call those breakpoints, and they were at 10.25 and 28.8 degrees. And we wanted to also look at the temperature range that we recorded in the Cow estuary, which was the minimum for when we're monitoring the movements. It was 11.4 and 28.4, which was pretty close to the breakpoints that were identified. So for my discussion, so we used the multi-method approach to, so two methods that we use to identify the habitat use patterns of these different mallee species. So otolith microchemistry and acoustic telemetry. So we're able to identify using otolith microchemistry that there was different patterns of movements that we have observed, 3 types of movements, and that could offer insights into first point, which was partial migration, where we had another type that was resident in the estuary, and then there was another type that was moving up and down, starting at sea and then moving into the estuary. And of those, as you can see from the graph, there were peaks from this graph here after they moved it into the estuary. So those peaks could be representative of the time where the fish are going to spawn. So if we compare with the resident type, it was in the estuary all the time. So some fishes will be going out to spawn, whereas others will remain in the estuary. And this could be a life history strategy that could be advantageous to the survival of the species. And then acoustic telemetry gave us fine-scale habitat use patterns whereby we're able to identify that there was some form of habitat partitioning between the species. So if we look at the two examples here, the striped mullet and the grooved mullet, these species were using different parts of the estuary. And we're also looking into what could be the drivers of such partitioning. So these things could be salinity. So we looked at literature where the striped mullet is more, has high affinity for saline waters than the grooved mullet, which could explain that distribution as well. And the last thing which we looked at was to look at how climate change might impact the movement of these species. So for example, what we found out from the acoustic telemetry data is that the grooved mullet did not use the upper reaches of the estuary during summer, where temperatures were close to their breakpoint of 28.8. But also summer, it was the time in which these species spawn. So using all of these methods to help us to understand how these species are moving, how they're using their habitats, and we can incorporate that into conservation and management for these species. Thank you.
Thank you, Bantini. Time for just one very quick question. Very quick one.
Hi, thanks, Anthony, for the lovely presentations. I love your graphics. Could it be also the other impact of your species movement could be the feeding behavior? And I also wonder, with the changing us not safeguarding our estuaries, I wonder what's the future for the movement of these species? Thank you.
Thank you, Alex. Yeah, so in terms of in terms of maybe the second, I'll tackle the second question. So climate change, we just looked at one species. So in terms of the movement during summer, where temperature in the rivers are elevated, they tend to move down into the lower parts of the estuary. So if temperatures theoretically continue to rise, that means there will be a restricted habitat for those species, which could limit, I guess, the resources that are available in the upper reaches. So yeah, that could be devastating for them. Yeah.
Bantini, unfortunately, we are ran out of time. Can we now move on to the next speaker? So moving from telemetry, we're moving back to videos and trying to see if these models can actually pick out fish. And we have Danielle Sally to present.
Hi, everyone. My name is Danielle Sali, and today I'll be presenting my paper on performance evaluation of YOLO V5, 8, and 9 in underwater fish detection. My supervisor is Dr. Dan Brown. So the study of fish populations and their intricate behaviors is fundamental to understanding and preserving marine ecosystems, with traditional capture methods relying on labor-intensive and invasive techniques, which are costly, time-consuming, and disruptive to marine life. Comes in a solution using underwater camera systems. One to be noted is weighted remote underwater video stations, which offer a non-invasive observation, enabling viewing of the fish behavior without disturbance But this comes with a caveat in which it generates large volumes of video data, with manual annotation being a significant time investment and often impractical due to the sheer volume of data, with one image taking up to 15 minutes or more to annotate. And a solution to this would be to implement an automated object detection system that can drastically reduce the time needed to do these annotations, and to allow for faster analysis, the creation of larger datasets, and comprehensive research, more comprehensive research. There are two primary methods for object detection. The first being a two-stage algorithm, which was found in the past to be slow, but accurate. This means that in the image, it identifies the region of interest first, and then performs the classification. Secondly, a one-stage algorithm, which does this simultaneously, reducing the time, so allowing for more real-time application, but in the past has seemed to be less accurate. But this has changed, with notably 3 articles mentioned there, where one-stage algorithms, notably YOLO v3, outperformed two-stage algorithm, first RCNN, by 6.4% MAP, 13.9% recall in this paper, showing also its real-time application with 20 FPS versus 14. Secondly, an article by Utsite et al., where YOLO v4, with its architectural advancements, improved upon YOLO v3 by 5.75% MAP. And the last article to show that context matters, just because there's a later version or more advanced architecture release does not mean it matches the context for underwater or the domain of underwater object detection. Therefore, these research papers need to be done to identify which architectural components fit the domain of underwater object detection best. One of the later versions of YOLO is version 9, which introduces a key innovation, solving the information bottleneck problem through the PGI, programmable gradient information, which means essentially is that information in the past was lost in the feed-forward process during the training. And this has been solved by introducing this component, and then also a more improved or efficient architecture through GLAN, with this potentially helping to improve accuracy and the efficiency of the challenging environments. So therefore, the aim of this study is to evaluate and compare the object detection performance of YOLO v5, 8, and 9 with a two-prong approach to this. First would be a benchmark dataset, benchmark experimentation with a homogeneous dataset, so traditional train validation test split. And this is done to establish a baseline performance for the models, but directly comparing them. And secondly, would be an out of distribution unseen test data set. So essentially, if a field trip was done in Seychelles, there will be many sites. Training on 3 sites and leaving 1 site out, the shift in water conditions or environment might play a role. And that's what this analysis is trying to see. And the expected outcome is to identify the strengths and weaknesses of these architectures, and also understand the impact of architectural advancement in object detection around water. So the first data set for the benchmark experimentation to be used would be DeepFish. This was a commonly used benchmark data set with 20 marine habitats along the coastal waters of tropical Australia. The capture method is cameras mounted on metal frames, which is deployed on the research vessel, and notably 15,463 instances or annotations. Secondly is a curated dataset by me in collaboration with SAIAB, which is the Seychelles 2020 dataset found in the Aldabra Island in the Indian Ocean. And this is a abrupt system dataset. And this achieved over 1 million annotations through semi-automated annotation and me correcting errors. This will be used for the benchmark experimentation. And lastly, the Seychelles reference library to see the out of distribution dataset context for analysis. So this is the same region of the Seychelles, but the reference library, and is done through scuba diving. This is the model information. So the large variance of all the models were chosen with those parameter sizes. Transfer learning was used from the COCO dataset. 300 epochs was used to run each model with the 640 by 640 image size. Those hyperparameters were chosen. and all the data sets were treated as a single class, so just detecting it as a fish, not species specific. And a standard chain validation test split of 70-20-10 was used. And each of these experimentations were done on the same machine for those specs. For the first experiment, for the benchmark dataset, YOLO V9 was a top performing in both deep fish and Seychelles, with notably in the deep fish having high precision, and in Seychelles both precision and recall as well as MEP. And this qualitatively shown here in these figures. With the first one, the left figure, showing that YOLO V9 got both the fish detections in such a difficult underwater condition. This can be seen as precision. And then your second figure showing that the other models V5 and V8 has done a missed detection, where YOLO V9 got all three from the ground truth. I don't know if you can see very well there. Sorry. And then for the Seychelles dataset, it had the highest recall as well, showing the difficulty in the dataset with the density of fish found in each image, with an average of 43 detections to a peak of 291 detections in a single image, where YOLO V9 also demonstrated the best performance in this dataset, which can notably be determined. The information bottleneck problem that was solved allowed it to learn features more effectively, therefore being the best performing model. In the generalization experiment, it's good to note that all the metrics dropped significantly from over 90s to 60s. This is showing the difficulty in an out of distribution dataset or more real world application, where these models perform well in the training or original analysis, but in real world application still struggles. And this needs to be analyzed further. So this can be shown in the qualitative analysis, where all the models did well in the foreground, but couldn't detect the smaller fish or the fish in the background due to the blue, possibly, that is brought through from the water. And this brings me to the conclusion. PGI is a component I seem to help in the underwater object detection domain. And it's evident that more generalization experiments need to be done so we can find it more applicable use in the real world. And the ways to do this would be to look at how to enhance feature learning. So there are various techniques, with the first being now looking at OBB orientated bounding boxes. So they fit more closely to the fish instead of just a normal box that takes in environment, or segmentation, semantic segmentation. Another way would be through data augmentation and synthesis, which would allow for more diverse feature learning, as well as to synthesize data with different environment or unseen environmental conditions so that it can overcome challenges with domain shift. And lastly would be architectural exploration. So to see which components fit the domain of underwater fish detection best, as shown before, because not the latest model always doesn't have the best performance. It's particular to a domain. Thank you. These are my references.
Thank you, Danielle. Unfortunately, we just ran out of time, so we'll move on to the next speaker. But please do engage with the speakers after, during lunch, and whenever you meet them. So our next speaker will be Dinam Kari, and she'll be speaking online. And again, it's on the theme of the telemetry and ecology of mullets.
Hello everyone, my name is Dinam Kari and I'll be talking about the movement and thermal tolerance of flathead mallets using a multi-method approach involving acoustic telemetry, otolith microchemistry, and ecophysiology. Estuaries are well known as important transition zones connecting both riverine and marine habitats, and these systems are highly productive and serve as necessary habitats for juvenile of estuarine-dependent species such as mallets. And my study focused on the flathead mallet, Mugelcephalus, which has a global distribution, dominates the coastal habitats, and is known to have a broad environmental tolerance. Although most studies exist globally about the species, not much is known about their movement ecology, habitat use, and importance of connectivity in South Africa. Flathead mallets in South Africa are important for aquaculture, subsistence, and recreational fisheries, and their current regulation for harvesting are set to 50 per person per day for subsistence and recreational fisheries, and unlimited for commercial fisheries, with no size limit or closed season. And due to this, the species may potentially face overexploitation. To properly manage this species, we need information on their movement ecology, which includes the drivers of movement such as temperature, their lifetime habitat use, which can be obtained from their otoliths, and their day-to-day movement patterns and space use, which can be obtained through acoustic telemetry. And through this, we will be able to understand the importance of riverine, estuarine, and marine connectivity throughout their life cycle. The first part of the project was to look at the lifetime habitat use of flathead mallets using otolith microchemistry. And for this, we used strontium and barium due to their correlation with salinity. And their chemical composition within the otolith were able to give us information about the natal origin and to look at variation in habitat use pattern within this population. From a total of 24 flathead mallets ranging from 0 to 5 years collected at the Kawi estuary, we were able to validate the marine signature by keeping them in the lab for a period of 2 months. After that, we extracted otoliths and used laser ablation to look at the concentration of strontium and barium within the otoliths. And then both confirmed that the beginning of life for the species is at sea. And we also compared the two elements and found that the barium was a better indicator of migration, as it is less influenced by temperature of the surrounding water and fish physiology. We were also able to identify 3 migratory groups using barium, and the first, which was the majority of fish, fell under the migratory group, which were the individuals that were moving between the estuary and the marine throughout their lifetime. And the second group, which was a bit smaller, consistent of estuary resident species, which are the species that skip spawning and remain in the estuary for the majority of their life. And then the last group was the marine residents, which were individuals that remained at sea for most part of their life. Moving on to the final resolution. Here we wanted to look at the home range and space use of this fish using acoustic telemetry. Here we wanted to look at the day-to-day habitat use, home range, residency index at different seasons, and connectivity of the marine, estuarine, and freshwater habitats. For this, we place multiple receivers in the Kawi estuary, which is in Port Alfred, and the receivers range from the mouth of the estuary all the way to the freshwater reaches of the estuary. And we caught and tagged fish with acoustic transmitters. A total of 21 fish ranging from 2 to 5 years were tagged with acoustic transmitters. And to look at their home range, the estuary was divided into 3 reaches. The upper reaches, which was closer to the riverine environment with salinity lowest, and the lower reaches, which were closer to the sea with the highest salinity. And the KDE home range estimates showed that the core areas were the upper and the lower reaches. The residency index, according to season, showed that all areas were used. However, the middle reaches were used more often across all seasons, and the upper reaches were avoided a bit more in winter compared to the other seasons. The individual fish plots shown here are an example of the day-to-day movement of flathead mallets in the Kawi estuary. I selected the 3 that left the estuary and made return trips. It's also important to note here that there's a decrease in the usage of the upper reaches in winter, which we will explore later. And finally, thermal experiments were conducted on this fish to look at the thermal limits of flathead mallets, since temperature is identified as one of the main drivers of species movement and distribution, and also to predict future persistence of the species as a result of impacts of climate change. And to do this, we looked at orbicular beats of fish over time, CT means, CT marks, to also determine the thermal range of the species. And for this part, we looked at 14 juveniles from the Kawi estuary to determine the CT mean, which averaged 3.5 and a CT max of 37.8, which we found that this was a very broad thermal range of the species. And even with minimum temperatures in Kawi reaching about 10 degrees, this is nowhere near their thermal limit. The main findings of the study was the clear evidence of migratory contingents in the population of flathead mallets that occur in the Kawi estuary. And this plasticity in life history has been reported in other marine fish species. And if we compare the two methods that were used here to look at the movement ecology, we saw that using otolith microchemistry, 60% of the individuals were migratory. However, when looking at acoustic telemetry, the results show that around 20% of individuals were migratory. This suggests the importance of using multi-method approach when looking at movement ecology, because acoustic telemetry only provided 1 year of data, and otolith microchemistry was able to provide a lifetime habitat use of the species. There are cost benefits related to migration. Migratory individuals have high access to food availability, leading to more growth and reproduction, and also high genetic dispersal, which is important for the persistence of the species, looking at the rescue effect. However, migratory individuals are susceptible to capture. The high capture rate of flathead mallets in the Kawi, we also observed it using acoustic telemetry, because from the tagged individuals, more than 50% at the end of our study period were removed from the estuary, with one of them being sold in a market in Durban, where we managed to retrieve the tag. In this population, there was also a small proportion of resident fish, both in the estuary and the marine environment. Telemetry results show that although some fish may be resident in the estuary, they still use the estuary lower, middle, and upper reaches extensively. And this may be due to the variable nature of the estuarine environment. Marine residents were also present in this population. However, these were underrepresented because sampling was only conducted in the estuary. And to further explore this, future studies will need to sample both the marine and estuarine environment to compare the contingencies. And finally, we saw the avoidance of flathead individuals in the upper reaches of the estuary in winter. Previous studies have shown that migration may also be thermal in nature, showing that fish leaving the upper reaches in winter might be related to temperature, although in the current study, we know that flathead reproduce in winter, so this might be coinciding with their spawning period. And the results from the thermal tolerance experiment show that this species can tolerate very low temperatures, although these experiments were conducted at salinity that was 35 parts per thousand, meaning that more experiments might be needed that combine both salinity and temperature effects on mallets. Thank you. And I would like to acknowledge the following organizations and individuals for their assistance during this study. And thank you for listening.
Thank you, Dina. Unfortunately, Dina is not available for questions and she's also out of time. So we'll move on to the next speaker, will be Ruth Matthews, who will be speaking about the usage of nursery habitats by spirid species.
Good afternoon, everyone. Again, my name is Ruth Matthews, and my MSc will be on the nursery habitat use by Boopsodia inonata, commonly known as France Madame, and the Spirodon devonensis, commonly known as the white mussel cracker. So firstly, nursery habitats are usually defined as areas that contribute a higher number of juveniles to adult populations relative to other habitats that may harbor these juveniles. And in order for these areas to be identified as nursery habitats, they need to have structure as well as food resources that result in the juvenile survival and growth, and that will allow for ontogenetic shifts of juveniles and subadults to their adult habitats. So the Spiridae family is a family of ray fin fishes commonly known as spirids or sea breams. And their major characteristics are that they're slow growing, late to mature, and some of them undergo sex change, which makes them very vulnerable to overexploitation. And South Africa is a hotspot for 42 species of these spirids. And 8.6 of these are highly threatened and recorded under the IUCN Red List. We see that a lot of these spirid species prefer nursery habitats in shallow coastal as well as estuarine habitats. And these are also important in South Africa. Examples of these are the blacktail, Cape stumpnose, and the straight-beak. However, very little attention has been given to marine spirids that only occur or use shallow rocky habitats as nursery areas. And examples of these are the white mussel cracker, as well as the France Madame. And we see that preliminary field observations have shown that the France Madame usually occurs in high rugosity sheltered reef and containing high coverage of red algae at flat rocks, Algoa Bay. And the white mussel cracker juveniles are dominant in the wave-exposed gullies west of the Cape Receipt. So a little bit about my species. The France Madame is small-bodied species that are slow to mature and long-lived. They are located in high and low reef areas for the adults and shallow subtidal reefs and gullies for the juveniles. The species is exceptionally important to the South African recreational and commercial fisheries in Eastern and Western Cape. However, very little information is known about the early life stages of this fish in terms of their movement, site fragility, residency, as well as the movement of subadults to adult habitats. The white mussel cracker is a much larger fish and a trophy fish for South Africa recreational shoreline fishery, as well as the spear fishery. However, their limited inshore distribution, large size, slow growth, late maturity, and longevity make them vulnerable to overexploitation in South Africa. And hence they have been listed as a near threatened species under the IUCN Red List. The juveniles for this species are highly resident to wave-exposed gullies. In order to quantify nursery habitat use, various methods have been used over the years, and these include methods like BRUVs or feeding ecology through stomach content analysis and acoustic telemetry, as well as tag and recapture. And my study will be focused on using feeding ecology, stomach content analysis, as well as acoustic telemetry. The main aim of my study would be to quantify the nursery habitat use of juvenile spirid species, the France Madame and white mussel cracker, within sheltered and wave-exposed shallow subtidal rocky habitats in Nelson Mandela Bay. And we will be quantifying these with the following objectives, which is the feeding ecology of the juveniles of both spirid species, juvenile species, in wave-exposed gullies with sandy bottoms and sheltered high rugosity reef dominated by red algae, as well as the nursery habitat use and preference of these juveniles in exposed gullies and sheltered high rugosity reefs. and it will be divided into two main data chapters. The first one focusing on the feeding ecology, and the second one focusing on the nursery habitat use as well as the preference of these juvenile species. And my study sites are Schoenmakers Corp as well as Flat Rocks. And the first two pictures above show a high rugosity as well as high wave rugosity, as well as exposed areas and gullies and rock pools. And the Flat Rocks area shows a much gently sloped area with high dominance of red algae. So the methodology for my chapter 1, basically, which is underway, basically involves capturing these marine juvenile sparrows using various capture methods. And after capture, they will be placed in sea-filled buckets where they will be anesthetized with an overdose of clove oil. And then they will be euthanized by placing on ice. And immediately after that will be transported to Ocean Science Campus at Nelson Mandela University. Here they will be measured for their total length, and specimen will be dissected in order to remove their stomach contents, which will be preserved, and to analyze the prey items in the stomach contents as well as their muscle tissue for stable isotopic analysis. Initially, for my chapter 2, we had explored the use of acoustic telemetry, which is a more accurate way of analyzing movement of target species in terms of their site fertility, as well as residency and their ontogenetic shifts to adult habitats through the communication between acoustic tags and their receivers. So basically, acoustic tags are surgically inserted into the juvenile fish, and they will release a unique signal, which is picked up by the acoustic receiver. Data can be picked up in intervals of times and recorded and analyzed. However, due to our preliminary results, due to results that have already been collected, we have found that the fish are too small to tag with the smallest tags. So we are now exploring the use of stereo remote underwater videos. And these will also answer our questions for movement in terms of their behavior analysis, which is the degree of their habitat use, their movement and feeding preference and site preference, and their relative abundance, as well as their ontogenetic movements with depth. And in terms of my outcomes for this study, I have already obtained my ethics in June of this year from Nelson Mandela University. And I would hope to graduate with my MSc and to publish my findings. But most importantly, I would like my work to contribute to recommendations and guidance for coastal conservation initiatives in terms of what quantifies good nursery habitats for juvenile sparrows that occur in shallow subtidal areas, and the movement and site fertility, as well as the ontogenetic shifts. To end off my presentation, I would like to end with the quote from Bowler and Benton 2005, which says, "The world's coastal environments are rapidly evolving. Thus, our foresight in preparing for such organismal ecosystem alterations is quite heavily dependent on the knowledge acquired from animal interactions, life history traits, physiology, and ecosystem services which facilitate species movement patterns." Thank you.
Thank you, Ruth, for the presentation. Any questions, suggestions? I think it's a proposal, so anyone has input?
This is a very difficult question to answer, but I'd like your thoughts on it. A lot of sparrows, like you showed, Rabdosarga salubi and others, use estuaries as significant nursery areas. but the two species that you're talking about here don't. Do you think it's because of the habitat that they use in the marine environment is not present in estuaries? Or is it the salinity tolerance of those two species is not conducive to them occupying estuaries? Or is it some other reason why we don't find those species using estuaries as nursery areas?
That is a difficult question. But I think we are looking more into that, and the whole point of the study is to find out what quantifies good nursery habitat for these two species. But we see that with preliminary studies, they are mostly abundant in rocky shores, and we're trying to find out what about these areas makes them more abundant there. And yeah. Thank you.
Can we give a round of applause? So we still stick to the same topic, moving to fish habitat associations in marine bay regions of Niger estuary. And to give this talk will be Luetu Mabaleka.
Hi, everyone. My name is Lutolwethu Mabaleka. My study is looking at fish habitat associations in the Marine Bay region of the Niger estuary. Coastal estuarine and marine ecosystems are highly productive, and they serve an important nursery function for our juvenile marine fish species. And within estuaries, there is a diversity of habitats which juvenile fish utilize as nursery areas. So the active utilization of estuarine habitats by our juvenile marine fish species is associated with the level of protection and it's associated with the availability of food, the reduced predation risks, and their structural complexity. In this regard, the high food availability and structural refuge provided to fish by structurally complex vegetated habitats leads to greater abundance and diversity of juvenile fish species, with these habitats referred to as high quality nursery habitats. The Niger estuary is the only natural estuarine bay in South Africa and is the largest estuary along the southern along the southern coastline. This estuary is recognized as one with the highest conservation importance and has the largest beds of seagrass Zostera capensis that is vulnerable, and has several other habitat types such as the rocky shores and several macrophyte species that include the seagrass Halophila ovalis and several macroalgal species, which are Calepa filiformis, Gelidium prestoides, Sergesum incestifolium, Plocamium corollariza, and has large areas of salt marsh. However, most studies on estuarine habitats and nursery function have focused on seagrass and salt marsh. However, the Niger estuary has a wide variety of other vegetated habitats with unknown nursery functions. And despite the ecological importance of our estuarine habitats, they are among the most threatened habitats. The threats to estuarine habitats include invasive species and anthropogenic threats. For example, in an Australian seagrass system, the green algae Calepa that looks like seagrass is taking over seagrass beds, yet its nursery function to fish is unknown. Similarly, in the Niger estuary, the invasive red algae Aspergopsis texiformis is now widely distributed and occurs within the seagrass beds. Additionally, large areas of seagrass Zostera capensis have been lost due to anthropogenic threats that include bait collection, nutrient pollution, and boating activities. And similarly, large areas of salt marsh have been lost due to development.
So a recent ecological study sampled fish throughout the Knysna estuary using seine nets and beam trawl nets to identify conservation priority areas. The findings of the study highlighted the Marine Bay and Lagoon regions for important fish species that are caught in the shore-based line fishery. However, these species include the Cape Town nose and the white string brass, just to mention a few. However, we do not know if these species are associated with specific necessary habitats in the Marine Bay and Lagoon regions of the Knysna estuary. So the aim of the present study is to examine the nature of habitat usage by juvenile fish in the Marine Bay and Lagoon regions of the Knysna estuary. To achieve this aim, we have two objectives that we're going to answer. The first one is to assess and determine habitat-specific fish assemblages in the Marine Bay and Lagoon regions based on species composition, relative abundance, and size structure. The second objective will be to measure and compare the necessary function of seagrass, Zostera capensis, and macroalgal beds for our juvenile fish species. So to answer objective one, which is to assess and determine habitat-specific fish assemblages in the Marine Bay and Lagoon regions. These are the sampling sites within the Marine Bay and Lagoon regions that we have selected. Sampling will be done seasonally in spring and summer, and our first sampling trip was in October 2024. So fish data was collected using seine netting. We also collected plant samples to determine the plant structural complexity indices in different habitats. And those complexity indices are canopy height and shoot density. We also collected the environmental data at the different sites using a YSI, and statistical analysis for our preliminary results were done on Excel and Primer. However, for further statistical analysis, we'll also incorporate RStudio as more sampling is scheduled for summer. So these are the sampling sites for our seine netting showed differences in their complexity indices. So we had short, dense seagrass, we had long, dense seagrass, long, sparse seagrass, and we had sand and a habitat with sand with free-floating seaweed. So looking at the fish community level, there were no significant groupings of fish in different habitats. However, at species level, we can see that certain species were more abundant in specific habitats. That is to mention the sauper and Diplodocus capensis, which would use seagrass and seaweeds. So they were also high in numbers at site 11, which was the sand with free-floating macroalgae. And Rhaptosaurus halobai, which has previously shown to select dense seagrass as their nurseries, was more abundant in site 9, which was the long, dense seagrass, Zostera capensis. So these species-specific results complement the results of previous studies that have been done on estuarine habitats. So the second objective will measure and compare the necessary function of seagrass, Zostera capensis, and macroalgal beds for juvenile fish, and fish data will be collected using stereo remote underwater video systems that are pre-calibrated. So for video analysis, we'll be using event measure and we'll identify fish to their lowest taxonomic level. And we'll also analyze fish behavior to determine the degree of habitat use. And then statistical analysis will be done on Primer and RStudio. So the data from this study will help determine key habitat hotspots within the Marine Bay and Lagoon regions, and thus identification of conservation priority areas within these regions. This information will aid in the conservation and management and restoration of high quality nursery habitats as means to replenish adult fish populations in the Knysna estuary. Thank you.
Thank you, Luto. Any questions for our presenter?
Something that you might want to consider is
the plant habitat like Zostra, for example, you may have a Zostra bed in the estuarine bay. It's not a marine bay, it's an estuarine bay. And then you may have a Zostra bed in the lagoon area, which is further upstream. But some fish species, for example, might tend to congregate nearer the mouth, like the Striopia that you're studying, whereas the Cape Stumpnose will occur much further up the estuary. So you might find the Zostra bed in the lagoon area being occupied by the Cape Stumpnose, and the Zostra bed nearer the mouth in the estuarine bay being occupied by the Striopia. So when you do your analyses on the use of the same habitat in different areas, be aware that fish species often are not evenly distributed throughout the estuary, and they may in fact be congregating in a particular area in a particular habitat simply because of the distance from the mouth, rather than the habitat per se. In other words, they're not occurring in the upper lagoon Zostra bed, but they are occurring in the estuarine bay Zostra bed. It's just something to consider.
Thank you very much. And just to add on what you just said, we actually did explore the NMDS plots, trying to find the species, to analyze the species abundance. We found that there were differences in our plots when we consider the region. So I'll just use what you've just mentioned to analyze what I see when I'm considering the regions. Thank you.
Once again, round of applause. We move on online again for our next speaker, Angus Zonwick, who will be bridging the gaps in underwater fish observation research, insights from a global questionnaire.
Good afternoon, everyone. My name is Angus van Wyk, and I'm a PhD student here at NRF-SAIAB. And today I'm going to be presenting a chapter for my PhD thesis titled Bridging Gaps in Underwater Fish Observation Research. These are insights from a global questionnaire. So the rationale behind this chapter stems from one of the aims of my research and thesis, which is to describe and to better understand the international network of users of underwater fish observation techniques. One of the ways we did this was through an epistemic analysis, looking at literature online for roughly the last 70 years to describe the research landscape and gain a formal understanding of the international network. The second objective was to circulate an international questionnaire to gain a contemporary understanding of what is going on with the international network, as well as to gain a deeper understanding. So just a brief introduction. When I discuss underwater fish observation research, I'm describing a suite of methods and technologies which collect observation data on fish using visual sensors underwater, such as cameras or divers. And being part of SAIAB, you may be familiar with a number of these techniques, such as stereo probes being used by MARIP or the ROV by ASAP. But there are a number of more traditional techniques, such as underwater visual sensors using scuba divers, and more modern techniques for technologies such as autonomous underwater vehicles. And these techniques as a research field collect incredibly useful information on fish species in a range of different habitats, from shallow water tropical reefs to deeper underwater canyons. And there are a few key points to make about underwater fish observation techniques from our previous research. The first being that although they are complementary in many ways, they do have their own suite of unique strengths, which are then leveraged differently by the community in different ways, such as baited remote underwater video systems for research on sharks, and underwater visual sensors and scuba diving surveys for coral reef systems. The second key point is the rapid expansion of the research field as a whole over the last 30 years, particularly driven through UVC research, underwater visual sensors, baited remote underwater video systems, and your ROV research. And the final key point is that this is a highly interconnected global community of both knowledge producers and knowledge users. And as one can imagine, with multiple methods, working in different habitats, and with an expansive growth of the community and it being a global community, there's a wealth of information and data being collected, and the power of which can be increased if the data can be made interoperable at larger scales, and if the network of users collaborated more effectively. So this is one of the primary functions of the consensus working group, which frames much of my PhD. And this is to make data interoperable, provide guidelines, and develop a community of best practice. So this is where the questionnaire and my chapter comes in. So we developed an interactive questionnaire online with the aim of collecting information on, simply put, who is in the community, how are they operating, why are they doing the research that they do, and what are the priorities and challenges that they face. So our research and the actual questionnaire is still live, but I thought it'd be great to present some of our more interesting preliminary findings. The first being how the dominance of UVC and browse as techniques mirror our previous research. We're also seeing a similar trend in the representation of countries involved in the research field. Although because we've had a series of local workshops here in South Africa, we've had high local engagement from our own networks there within South Africa and the Seychelles. And what we continue to see when we look at the international network as a whole is how diverse we are as a community. So we are made up of an array of different sectors, although at the moment anyways, it's primarily academic. We work in almost all oceans, although this does seem to be driven a lot by the WIO, or West Indian Ocean. And as a research community, we operate in almost all environments, although so far our efforts seem to be skewed more towards shallow water environments or shallower water environments, this reinforcing the need to continue efforts in understanding our deeper water environments. So one of both consensus's goals and of the questionnaire was to gain a better understanding of standardization within the community and what really drives adherence to best practices and the willingness to follow standard operating procedures. Well, as it seems so far is that the vast majority do follow best practices and are willing to do so, with the primary reason those who don't being simply that they just don't know, which is a somewhat solvable challenge. But other factors that contribute to adherence are experience. So the longer a researcher has been in the field, the higher the chances are that they conform to best practices, and that your economic status may be linked to this adherence as well, with developing nations showing higher adherence, perhaps driven by a reliance on established methods. And that connectedness within the network, so stronger collaboration ties, also aligns with strong adherence to best practices. And lastly, that it's not uniform amongst methods. So some methods do conform higher than others, such as within UVC and browse, as opposed to towed underwater video systems, which had a so far 100% non-adherence to best practices. So given the importance of the perception of connectedness within the network on your willingness to adhere to SOPs and best practices, we thought it'd be very interesting to dig a little deeper into this perception of connectedness. So both at a national level and at a international level. And what we're seeing is that generally speaking, people feel fairly connected within their national networks. But this starts to break down when you look at it as an international network. What's even a little more interesting here is when you split this data up into your developed and developing nations, sort of falls away. And what you're seeing is that, once again, there is some degree of geographical bias, with the developed world feeling far more connected to the international network, and with your developing nations feeling less connected, some degree more in more of a silo to some degree. So lastly, we had a section in the questionnaire with open-ended questions to understand the priorities of the community based on themes, what their goals are, and what their primary challenges are to achieving those goals, or their priorities. So 4 key themes that came out were large-scale distribution patterns of species were a priority, the effectiveness of marine protected areas, the effects of climate change, and lastly, the desire for the standardization of methods at larger scales. And this sort of aligns with some of the goals that were highlighted. So informing policy and management being one of the primary drivers within the research field, supporting conservation efforts, monitoring, and being quite a technology-heavy research field, obviously advancing scientific knowledge being one of those. So this comes as no surprise when we discuss challenges, particularly around the number of workshops we've had this year, but funding remains the biggest challenge for the researchers and users to achieving their goals or priorities. This is part about logistics and equipment availability and the challenge of working on the water. So this is just a snapshot in 8 minutes of some of our results, but what it's giving us is a strong data-driven platform from which we can create a framework to strengthen the network and mobilize the knowledge created by the community. So focusing on emerging scientists, for example, strengthening international networks, and ultimately monitoring connectivity as a metric to gauge the effectiveness of our efforts. And developing this framework and applying it are our next steps. So I look forward to presenting these findings to you next year. Thank you so much.
Thank you. I guess we'll have time for just one quick question, and I think Angus is available for answering. Okay, seems like there are no questions from the floor. So in the interest of time, I think we will just move on to the next speaker. So our next speaker is Opemetse Mokwa, will be speaking on the genetic and morphological differences between eelgrass and the chrome estuary. And this presentation is also online.
Good day to all. My name is Opemetse Mokwa, and I'll be presenting the first data chapter of my master's thesis titled Genetics, Morphology, and Resource Availability of Two Morphologies of Eelgrass in Crom Estuary, South Africa. I was working under the supervision of Professor Rajkaran, Professor James, and Dr. Mecha. Seagrass beds have long been recognized as some of the Earth's most productive and valuable ecosystems. They perform many ecosystem functions, including filtering water, stabilizing sediments, and further preventing erosion. The dense meadows can also enhance carbon sequestration. Studies have shown how seagrass meadows are important nursery areas through the provisioning of food and habitat to fish and invertebrates. They also contribute to recreational, economic, and subsistence value of coastal ecosystems. However, even though seagrasses are widely studied, they continue to be threatened globally by climate change impacts such as increases in marine heat waves and storm surges, with anthropogenic impacts including destructive fishing practices, recreational activities, as well as eutrophication. Our study is based on Zostera capensis from the family Zosteraceae. This family only has 3 genera, and the genus Zostera has 15 accepted species. Zostera capensis, which is commonly known as the Cape Dwarf eelgrass, is the only species of Zostera found on the tropical and temperate regions of Africa. It is also the only dominant seagrass in South Africa. It is popularly known for its ecosystem engineering capabilities, as they are able to create complex habitats and structurally simple ecosystems. In South Africa, this species continues to be lost in estuaries because of both climate change and anthropogenic impacts, and as a result, its recent classification was changed from vulnerable to endangered, calling for efforts towards its restoration and conservation. The problem identified is that populations of Zostera capensis show population differentiation with 2 lineages in South Africa, and in Crom Estuary, 2 discernible forms of Zostera, the short form and the long form, were observed. The aim of the study was then to investigate whether these 2 morphologies of Zostera are distinct forms or different native species. The objectives were to quantify morphological characteristics and compare the 2 morphologies, while the second one was to carry out a molecular phylogenetic analysis using RBCL and MEDK chloroplast regions. Our study site was in Crom Estuary in St. Francis Bay in the coast of Eastern Cape, South Africa. The following methods were conducted to reach our objectives. For morphometrics, seagrass metrics were measured between the 2 forms of Zostera capensis, and these included leaf lengths, width, rhizometrics, as well as density. Below and above ground biomass was measured in the lab. For genetics, we extracted DNA from fresh material collected from the estuary, and we amplified gene regions like RBCL and MEDK. The sequences were then aligned and a tree was computed from Mega. For statistical analysis, we used the R software. There was variation in the morphology of the two forms, where compared to Form 2, Form 1 had a shorter leaf height, narrower leaves, presence of a red longitudinal vein, longer internode length, as well as shorter roots. The morphometric parameters of Zostera capensis in the CROM showed significant variation across the two forms, which likely reflected phenotypic differences in the seagrass beds within the estuary. On average, Form 1 had a shorter leaf height, ranging about 7.9 centimeters, which is an unusual observation for Zostera capensis during the summer. This can be due to morphological acclimation, which was also observed in Langevan Lagoon, where there were two morphotypes, the large-leaved one ranging from about 20 centimeters and the small-leaved one with about 8.6 centimeters. Other possible explanations for the differences in morphology included heat stress, where the reduced leaf length and shoot cover in Form 1 could be resulted to heat stress, as observed by Bergman et al. This is because high temperatures can impair the plant's ability to photosynthesize by damaging the chloroplast, reducing the energy production needed for growth. High densities are then followed by a rapid reduction in shoot cover, the likely cause of which is heat stress, resulting in the loss of shoots and leaves. Marsh et al. Observed a similar trend in Zostera marina, implying that a negative carbon balance and potential limitations on carbon uptake lead to reduced growth rates in Zostera species. To complement the results obtained from the morphometrics, the RBCL sequences were used to compute a maximum likelihood tree as seen above. The Form 1 samples appear to be closely related to Zostera micronata instead of the samples from the known Zostera capensis. The sequences recovered suggest that the two forms may not be different species, but two morphotypes that may have resulted due to phenotypic plasticity, mirroring the studies by Tanaka and Nakaoka, as well as that of Shafa et al. In conclusion, it is important that we understand the population dynamics of Zostera capensis, as it offers resources to a variety of organisms in the estuary. The presence of the smaller morphotype may result in a reduction of ecosystem functioning of the meadow, further reducing biodiversity in the estuary. I would like to thank the University of the Western Cape for allowing me to do my master's at their institution, the NRF-SAIAB for allowing me to use their lab, and together with the Mastercard Foundation Scholarship for funding my studies. I'd also like to thank the name listed above for their contribution towards my data collection as well as my data analyses. Lastly, I'd like to thank you all for listening to my talk.
Thank you. I would also like to thank you for keeping it very short. We have time for a few questions. Yes.
My observations over the years with the eelgrass in estuaries is that the long-leafed form, which is also broader, has a broader leaf as you described, is normally in the subtidal areas of estuaries. In other words, even at low tide, it is still fully inundated with water, even at a spring low tide. Whereas the shorter leafed, narrow leafed version is normally in the shallow intertidal area and can be exposed for quite considerable periods, particularly during spring tides. And in summer, with the heat, that is probably less vulnerable to desiccation and to heat from the sun than would be the long leaf version, which is broader, which would be more vulnerable. Do you think that is a valid observation or not?
Thank you for the question. I actually do think that is a valuable observation. However, we are still trying to understand whether is it because of the heat stress and the temperature, or it's just any other different species that may have invaded the estuary.
Good. On the floor. Okay, I think we can move on. Thank you very much for the talk. So the next speaker is Esihle Deleni, and she will be updating the checklist of freshwater fishes in the Kruger National Park and trying to answer the question, what lies beneath?
Thank you. Good afternoon, everyone. I'll be talking about updating the freshwater species checklist in Kruger National Park, including ephemerals and isolated water of the Nkomati catchment. Okay. This study is based on the freshwater fishes of Kruger, as I've mentioned, based on the publication by Vida V. Pinar in 1968. Pinar was a sandpack scientist based in Kruger National Park. He believed that fish species, just like any other mammals, reptiles, and birds, also amphibians in the park, needed to be known. He aimed to show the diverse forms of freshwater fish species in Kruger National Park, as he believed they were an important component of the national biome. He did the study in the Limpopo system and the Nkomati system, which covers the north and the southern part of the park. He discovered communities and species diversity of fishes such as Glossogobius, Garis, and Cerochromis meridianus. Since then, considerable research in the park has been done, but this research in rivers have focused on the larger rivers such as the Limpopo River and the Crocodile River, and in terrestrial animals, but not much on the small pools, dams, and ephemerals of the park, which received relatively little attention. Atlantic waters across the park have been considered almost exclusively through their lens of the use of terrestrial animals, whereas these water bodies are still intact freshwater habitats, and the focus on these ecosystems need to be maintained, although there have been threats such as invasive species and floods in the park. So the aim of the study is to update the freshwater fish species checklist in Nkomati catchment using historical data and also ongoing species surveys using morphological data and DNA barcoding. This aim was motivated by a review by Russell in 2011, where he says big national parks such as Kruger National Park should focus on ephemerals and conserve them. The first objective of the study is to identify communities and organisms of ecological interest, rarity, vulnerability, and inform potential management priorities, and also to examine the species diversity and distribution in the seasonal rivers and dams. The study will take place in Kruger National Park. It is a national park with the savanna biome and has semi-arid regions. We will focus on the southern part of the park, which is the Nkomati catchment. The Nkomati catchment runs through the park from Mozambique, and it breaks down into various tributaries and isolated waters. We will sample using seine nets, fag nets, traps, backpack electrofishing, throw nets, and also collect water parameters with multi-parameter. After sampling, we will use the freshwater fishes of Southern Africa guide by Paul Skelton 2024 for morphological data and bring back data sampling tissues back to the lab. What we want to do is to create a species checklist and also have the data material to create phylogenetic trees so that to complement the morphological data. We hope to find species such as Cacha brevis, Enteromias brevipennis, and Leobarbus nespretensis, as these species are some of the species that are found mainly in the Nkomati catchment. So as our outcomes, we would like to generate a novel and substantial records of fish species in the system that will work as a baseline for future reference and comparison with the historical data to identify rare and threatened species and focus on management efforts on these respects. And also to create a curated poster of freshwater fishes of Kruger National Park and where to find them to be displayed for tourists. We would like to get all the data in the Nkomati catchment for voucher specimens and genetic materials for SAIAB and also SANParks Kruger biodiversity collection. Thank you.
Thank you, Cecilia. We do have time for a few questions. Okay, whilst people are thinking, I have a question for you. In order to raise like awareness of the fishes and the Kruger and all these inland fishes, do you think there will be any benefit in having something similar to the, we have large aquariums for marine species, but having like a aquarium for something like that in the Kruger, do you think that would help people be more aware of what's beneath the brown waters in the rivers?
Yes, I think that would benefit not just Kruger alone, but like raise awareness for educational purposes as well. I remember telling someone what my research is about, and his question was, do Kruger have fishes? So I think that will raise a lot of awareness. And also, if you go to Kruger as a tourist, you'll find a lot of handbooks on birds, amphibians, reptiles, mammals, but nothing on the fish that occur there.
Any further questions from the floor? Okay, SC will be around. Please do ask her questions and she'll be ready to answer those. Moving on to the next speaker. So we have Nontebo who will be speaking on the biodiversity assessment of Mpumalanga high-fault wetland systems. And this is an online talk.
Afternoon, everyone. My name is Nontebo Leshaba, and today I'll be presenting my PhD research on biodiversity assessment in the Mpumalanga high-fault wetland systems to come up with strategies for effective conservation and biodiversity of SERTs. To give a little bit of background on wetlands, the pictures depicted on the slides show different types of wetlands that are formed through different processes. These are transitional land between the terrestrial and aquatic systems. They can either be seasonally or permanently saturated with water. However, my study focuses on a type of wetland that is called a pan-wetland. This type of wetlands, they depend on continuous supply of groundwater or rainfall for their composition, structure, and functionality. They are important ecological systems providing a range of supporting, cultural provisioning, and regulating services. However, despite the above-mentioned importance, these wetlands are still critically endangered due to the land use activities, more especially in my study area, where there's crop production and livestock farming. These activities alters the ecosystem functioning and has an impact on water quality, which also influences the biodiversity within our ponds. Hence, the study's overall aim is to enhance the biodiversity assessments of these wetlands by integrating the eDNA techniques with our traditional ecological surveys. We also seek to evaluate the impact of land use on these wetlands ecosystem functioning and further investigate the effectiveness of our biodiversity offsets in an agricultural landscape, and to also look into the adequacy of policy frameworks that are there for wetlands conservation and restoration. As depicted on our slides, we have the set of objectives. Number one, we want to quantify invertebrates biodiversity using eDNA and traditional methods to enhance biodiversity assessments. We also want to assess the effects of land use activities on the nutrient cycling, chlorophyll A concentration, and water quality. We further want to evaluate the effectiveness of biodiversity offsets on wetland across an agricultural landscape and examine the existing policy frameworks governing biodiversity conservation and wetland restoration. This will help us to assess their effectiveness in addressing current challenges in order to achieve conservation goals. This research takes place in the Mpumalanga Lake District, with its focal point being the village of Chrisismia. The Chrisis system accommodates a diverse range of ponds, and it is located at an elevation of 1,707.39 meters above sea level. It has tropical climate with dry winters. This is according to the CWB classification. The district has an annual temperature of 23.16 degrees Celsius, which is 1.94% higher than the South African average. It also receives an approximately 74.36 millimeters of precipitation and has 124.53 wet days yearly. Looking at our site selection, we have 3 pan wetlands that are located on private farms dealing with crop and sheep farming. At each pan, we have 3 sampling sites for both eDNA and macroinvertebrate sampling. For eDNA sampling, 500 milliliter bottles will be used to collect water samples, and this will be filtered using a hand pump on site or in the lab, and analyzed using a CPR with COI primer for detecting macroinverts. For traditional methods, we'll be using a kick sampling, which uses a handheld kick net to collect the inverts, and they will be preserved in a 70% ethanol, and their identification will be done under microscope up to genus level. Moreover, we will be collecting data for water chemistry, looking at water quality parameters such as dissolved oxygen, total dissolved solids, salinity, pH, and more, which will be measured in situ using a multi-parameter meter. Then nutrients such as phosphate, nitrate, and ammonia will also be analyzed, and chlorophyll A concentration will be analyzed from the collected water samples as well. Then to evaluate the biodiversity offsets, we'll have a baseline assessment of the existing wetland biodiversity and ecological conditions. Then we'll use field surveys such as species inventory and habitat quality assessment. We also look into metrics such as vegetation structure, water quality, and the presence of indicator species, and use some historical data and remote sensing techniques. All this will help us to gauge the success of the offsets. Then for the evaluation of policy frameworks, we'll develop a search string and gather studies from databases such as your Google Scholar, Scopus, and more. That focuses on policies related to biodiversity and wetland restoration. Then we'll use a thematic analysis to identify gaps in the current frameworks and suggest improvement based on the study findings. For data analysis, we'll be looking at different methods. In the macroinvertebrates community analysis, we look at diversity indices, and we'll run some statistical tests like the Kruskal-Willis test to check for the differences in diversity across ponds and seasons. We'll also look into anonism just to test the taxa composition and environmental differences. Then for eDNA, we'll be looking at DNA meta barcoding, then environmental influence on the communities of the inverts. Will be using a CCA just to examine the relationship between inverts and the environmental variable. And to gauge the offsets, the biodiversity offsets, we'll be using descriptive statistics, comparative tests, multivariate techniques, regression models, spatial analysis on QGIS or ArcGIS, and look at temporal trends using the time series analysis and repeated measures on ANOVA. And lastly, for policy framework analysis, I'll be looking at thematic analysis, or I'll be using thematic analysis by Brown and Clark. 2006, which uses a 6-step approach. And this will be coded in a software called the Atlas TI 2022. And this method helps in the identification of themes and patterns from a qualitative data. Thank you so much.
Thank you. Unfortunately, Nompumelelo is not online to answer any questions. So this rounds off the last talk for this session. So can we just give all the speakers a round of applause? We can now break for lunch. I kindly ask you to be back here at 2:10. Thank you.